acknowledge our session in charge, Dr. Guru Murthy ji, for his guidance and support in shaping this session.
We would like the session chairs to kindly take the dais, please. Thank you, sir. Thank you, ma'am.
I would like to special welcome to our distinguished, invited and plenary speakers, respected faculty members, students, researchers, and joining us today. Your presence truly enriches this platform for knowledge exchange and collaboration. This conference, organized by Amity University, Uttar Pradesh, in association with the Biotech Research Society of India, International Bioprocessing Association, and the Center for Energy, Environment and Sustainability, with Unipic and IYouth as the gifting partners, stands as a testament to our shared commitment towards sustainable biotechnology and bio-innovations. Today's program is designed to be both engaging and impactful, featuring insightful keynote lectures, thought-provoking presentations, and interactive sessions. We encourage all presenters to please keep their talks clear, focused, and time-bound to ensure an enriching experience for everyone present here.
With that, let us begin today the proceedings. I now have the honor of introducing our first session chair, Dr. Deepak Ganjewala ji. Dr. Deepak Ganjewala ji is currently professor at Amity Institute of Biotechnology, and also he is the center head of Center for Plant and Environmental Biotech, AIB, Amity University, Noida. He has distinguished career both as an educator and researcher in the field of plant biochemistry and biotechnology. With nearly 25 years of experience in higher education, research, and mentoring students, he has established himself as a leading and researcher. As pioneer researcher, he made significant contributions to plant secondary metabolism, mainly essential oil biosynthesis and regulation in lemongrass. He also works on bioprospecting and microbial biotransformation of secondary metabolites. He has published more than 90 research papers in esteemed journals, including book chapters and also a book. He has successfully completed several research projects sponsored by national funding agencies. In recognition of his achievements, he received the Distinguished Scientist Award from the Society for Plant Research in 2024. Furthermore, he is the recipient of the Israel Government Scholarship, DST Young Scientist Award, and Summer Research Fellowship of the Indian Academy of Sciences, India. May we have a huge round of applause for our session chair.
Okay. So now I would like to introduce our next session chair, Dr. Swati Sharma ji. She is an assistant professor in Department of Biosciences at Integral University, Lucknow, with specialization in botany, microbiology, plant microbe interaction, plant pathology, and post-harvest technology. She holds a PhD in botany and has a significant research experience from her work as a CSIR NET qualified researcher and SRF that focused on the biological control of plant pathogens and eco-friendly disease management strategies. At Integral University, she teaches postgraduate and undergraduate courses in microbiology and biotechnology and supervises various research projects that includes the plant growth promotion and bacterial bioformulations. She is also involving in guiding PhD students and master students, and her records includes numerous publications in international journals, book chapters, contribution to research on microbial technology, plant disease control, and post-harvest biology that reflects her commitment to sustainable agriculture and microbial biotechnological research. Please put your hand together to
With this, let's start today's session. And I invite our session chairs to kindly take their chairs so that we can start with the session. We welcome you, sir and ma'am, and we hand over the mic to you now, sir.
First of all, I would like to invite Dr. Bhanu Revati K, Dayanand Sagar College of Engineering, and this will be a lecture on bioremediation of nickel toxicity in water. So I'd like to invite Dr. Bhanu Revati K, Dayanand Sagar College of Engineering, and it will be a lecture on bioremediation of nickel toxicity in water and effect of nickel-induced oxidative stress in SHSY-FY cell lines.
A very good morning to the session chairs and all the distinguished faculty of Amity University and all dear participants. I am Bhanu Revati K and I am an assistant professor at Dayanand Sagar College of Engineering. I am from Bangalore and I will be presenting a part of my research work, which is bioremediation of nickel toxicity in water and the nickel-induced neurotoxicity in human cell lines of SHSY5Y.
नेक्स्ट तो होता ही है शेयर करो करती एग्जिट करनी तो बंद हो गया मैम हो गया शेयर करो जी करियो हो गया अब आप उसमें चेक करो लीजिए मैम सॉरी फॉर द
Extremely sorry for the inconvenience. It's not happening.
इस तरीके से अभी भी हो रहा है डुप्लीकेट बटा एक मिनट रुको डुप्लीकेट पे डाल दी एक मिनट रुको ओके करो क्लोज करो डिस्कनेक्ट हो गया ठीक है, अभी से 1 मिनट रुको। अब नेक्स्ट बोल रहा था इसे भी कभी भी रूम पे तो शेयर करो। ये शेयर करो, ठीक है, देखना इधर देखो एक। अरे ऑलरेडी हो रखी है भाई, वो दूसरी लड़का। इसमें नहीं डाल, डुप्लीकेट पे आपके सामने तो डाला था, हम डाल। पर वो स्लाइड नेक्स्ट स्टैंड पे मत, from this one for next, this was next, ये क्या कुछ है, रेड कर दो उसको।
Extremely sorry for the inconvenience. So this is the topic of my presentation, and
Yes, now it, and this one for next.
So I'll be talking about the toxicity of nickel in water. Nickel is a heavy metal and it is not allowed beyond 0.02 mg per liter in drinking water according to FDA guidelines. And according to the Environmental Protection Agency guidelines, it's not supposed to be more than 0.1 mg per liter in the environment. So nickel is an element, it cannot be degraded into any other form. So it has to be chelated and it has to be removed from the source of contamination. So it mainly enters through electroplating industry and other forms of man-made activities. So when it enters water and through water, when it enters various ecosystems like marine ecosystem or the plants, in plants it causes it causes increased oxidative stress. It causes increase of reactive oxygen species in plants and animals upon ingestion, and it increases necrosis and chlorosis and also manifests in decreased photosynthesis. In humans also, it is found to cause these effects. So it increases activated phagocytosis. It releases various reactive oxygen species such as the superoxide anions and also the hydrogen peroxide and other peroxide related anions, which cause increased oxidative stress, and thereby it increases lipid peroxidation, that is the oxidation of cellular lipids and also cellular proteins and also DNA damage. So it also was found to cross the blood-brain barrier in humans upon acute and chronic toxicity, and also causes various other forms of toxicity in humans. So my objectives of research are to assess the bioabsorption capacity of two important plant-based biosorbents, Bacopa monnieri, also known as Brahmi, and also Moringa oleifera, the common drumstick or horseradish as we use it in edible forms. So I'll be taking the leaves of these two plants and assessing their bioabsorption capacity of with nickel in the solution form. And also my second objective is to assess the bioabsorption potential through adsorption isotherms, whether it is thermodynamically feasible, and also have evaluated the neurotoxicity of nickel and the reduction of this toxicity through these two plant extracts in the human neuroblastoma cell line SHSY5I. So this was the methodology that I followed. First, I had taken the biosorbents, prepared them, and then sieved them to make them very particulate, and assessed the functional groups through FTIR analysis. I have optimized the physicochemical parameters required for biosorption, such as pH, temperature, the dosage time required for biosorption, and also the contact time between the sorbent and the sorbate. And after removing the plant biosorbents, I had subjected them to colorimetric estimation of nickel, and later I had plotted the data into Langmuir and Temkin adsorption isotherms, and also tried to see whether it fits into the Gibbs free energy equation. Delta G is equal to delta G is equal to minus RT ln KD. So if delta G is negative, you know that the reaction is spontaneous and feasible. If the delta G is positive, the reaction is non-spontaneous. So also I had assessed the neurotoxicity of nickel by exposing them to SHSY5Y neuroblastoma cell lines. These are widely used for environmental toxicity studies. So I had estimated the LD50 of nickel and also the plant extracts. And also I had seen the bioabsorption parameters, that is, sorry, the oxidative stress parameters, that is the enzymes superoxide and dismutase and catalase are the enzymes that are upregulated during oxidative stress. It is a defense mechanism of the cells to reduce oxidative stress. And these two marked in red, the protein carbonyls and the lipid peroxides, are actually the oxidized products or the damaged products of the cells after oxidative damage. So these are the results of my findings. So FTIR spectrum indicates the peaks at 3,400 to 2,800 nanometers for Moringa oleifera as well as for Bacopa monnieri, indicating that it has negatively charged functional groups such as OH and carboxyl groups. So I saw that the highest bioabsorption occurred at pH between 6.6 to 8 and the temperature of 60 degrees Celsius. The bioabsorption dosage for optimum bioabsorption was found to be somewhere in between 0.8 to 1.2 grams per liter of the solution. And also the contact time required for optimum bioabsorption was 50 minutes. So beyond this, I had fitted the data into Langmuir isotherm, which is generally followed by most cations. And Langmuir isotherm showed promising results from the R-square value, as you can see from the graph. And I had calculated the Langmuir isotherm constants. Temkin isotherm basically talks about whether the binding energy is enough to cause physisorption or chemisorption. So basically it talks about the thermodynamic feasibility of biosorption. And also I had fitted the data into the Gibbs free energy to see the thermodynamic feasibility. So generally a positive slope indicates that delta G would be negative. And you can see from these two graphs that we got the positive slope. However, the R-square value was a little reduced. So positive slope indicates that the process is actually thermodynamically feasible. So upon exposure to various concentration of nickel through MTT assay, I was able to find out the LD50 value of nickel as well as the LD50 value of Bacopa and Moringa using AAT BioQuest calculator as an online tool. And in SHSY5Y cell lines, you can see before and after treatment that the cells have reduced, the contact reduced, and also the adhesion between the cells also was interfered due to exposure to nickel. So these were the results of catalase superoxide dismutase assays. So these two enzymes are supposed to be elevated, but unfortunately, when the cells were exposed to nickel, these two enzymes were found to be inhibited. So is the case with most of the heavy metals. So these heavy metals actually inhibit the levels of superoxide dismutase and catalase. So I had taken 1/10 of the LD50 concentration and exposed the cells to these concentration. And as you can see, alone, Bacopa and Moringa did not enhance the activity of catalase and superoxide dismutase to a greater extent. But when prophylactically exposed to these plant extracts before exposing them to nickel, you can see that the levels of these two enzymes increased significantly. Especially Bacopa was very monumental in increasing the levels of catalase and superoxide dismutase, because of which the cells will be able to combat the oxidative stress. So malone dialdehydes and protein carbonyls are actually the damaged oxidative products of lipid peroxidation and protein carbonylation. You can see that the plant extracts have considerably reduced these two oxidized products or oxidized byproducts or the damaged products. So these are my conclusions. So the first two in the table, QM and B, are the Langmuir constants. So higher Langmuir constants indicate higher value of QM indicates higher ability to biosorb, and B actually indicates the Langmuir biosorption coefficient. So higher coefficient means that the affinity is more. So it actually obeys Langmuir constant, which is like an Langmuir kind of isotherms, which is like an enzyme and substrate. And also the KT value actually indicates the affinity or the physisorption or biosorption or chemisorption process. So a value of Kt below 1 indicates physisorption and a value of Kt above 1 indicates chemisorption. So you can see from the table that Bacopa follows physisorption, whereas Moringa oleifera follows chemisorption for the uptake of the heavy metal. So the thermodynamics were evaluated. The delta G was found to be less than 0, which indicates spontaneous and feasible reaction for both the biosorbents. And the enzymes were found to be elevated upon prophylactic exposure to these plant extracts, and the lipid peroxidation and protein carbonyls were found to be reduced. So these are my references. Thank you for your patient listening.
Yeah, it was very nice presentation and nice work you have done.
Thank you, sir.
We have like few questions.
Sure, sir.
Bacopa monnieri and the Moringa oleifera, but they are known for what kind of bioactive properties?
So Bacopa is known to be a neuroprotective compound. It has been studied to reverse aging and also neurological disorders. There were lots of studies that it has effect in reducing oxidative stress and neurodegeneration and all those things. So similarly, Moringa, actually, it is very well known to reduce oxidative stress. It is an anti-inflammatory, and also Moringa is known as a super adsorbent. It's known as a clarifier. So it is very well known for absorbing heavy metals from the solutions.
So one more thing I want to ask, that how did you measure the carbonyl
Content.
content?
So the protocol was from Levin et al. So basically, protein carbonyls are actually adducts that are formed. And when they react at a pH of 8 in the phosphate buffer with DNPH, dinitrophenyl hydrazine, they actually emit out a color. So the reaction was carried out at dark, and then the absorbance was taken at 432 nanometers.
So what extent you have found the damage to the protein?
Sir, actually the damage to the proteins was very high when it was treated alone with nickel. So if you see the results here, so nickel actually increases the protein damage. You can see that the protein carbonyls indicated in orange is very high. So if the plant extracts are able to lower this protein damage, it would be a very significant result. So I had seen that the protein damage was reduced at least by 25% to 30% with Bacopa and Moringa. So with Moringa, the extent of amelioration is really high.
Okay, thank you. Now the talk is open for the further questions from the audience as well. If audience have questions, so we can take one or two questions. Okay, I think there are no questions. So thank you so much, Dr. Bhanu.
Thank you, sir.
Okay, shall we invite the next speaker? Okay. So now we move to the next speaker, Dr. Yashika Raheja. She is from Guru Gobind Singh Indraprasth University, and her topic is bioprospecting thermophilic fungi for developing efficient cellulose cocktail in hydrolysis biorefinery relevant pre-treated lignocellulose. So we invite Dr. Yashika to present her work.
A very good afternoon to all of you. My name is Yashika, and today I will be talking about developing thermophilic fungal strain Rasmussonia emersoni and the cocktails for efficient hydrolysis of biomass, pre-treated biomass. So, okay, thank you. So the major motivation behind this work is, as we all know, that in India we generate a lot of waste, that is lignocellulosic biomass, and our government, they have majorly focused on converting these waste into value-added chemicals and fuels. So one of the major fuel is the bioethanol, where the government has achieved the blending of 20% up till now. And the major industries in those ventures include the Paraj Industries, Indian Oil Corporation, and Panipat biorefineries. But I must tell that this process is not that easy. There are several technological challenges associated with process related to capital expenditure, operational expenditure, maybe the biomass pre-treatment cost, enzymatic hydrolysis. So if we talk about today's market, so the Novozymes, they are the major enzyme manufacturer that has launched Selexitec 3 as a commercial cellulase. But this commercial cellulase, although it gives high saccharification efficacy, but the protein loading rate for these cellulase is quite high. So we have tried to address one of the challenge, that is this cost associated with the cocktail. So we in our study have developed a cocktail that is competitive to this commercial cocktail, and we have tried to reduce the load as well as increase the saccharification efficacy. So I'll start with my work. So this is the overview of my work. Here we have selected, if you see, Rasmussonia emersoni. This is a thermophilic fungal strain. So we have developed this strain within a series of steps, and after development, the main goal which I mentioned is to develop a cocktail. That is, we have developed the Ramazyme named cocktail. So I will tell how we have developed this strain. So firstly, we have evaluated the cellulolytic potential of this strain. For that, we have carried out submerged fermentation, and we have taken several substrates you listed over here. And we found that the enzyme production was higher on the chickpea flour based medium. So as we can see clearly, the activities indicating the endoglucanase, cellobiohydrolase, beta glucosidase, they are quite high at using this substrate. So under these conditions, we have tried to evaluate the secretome of this strain. So where we found that the glycosyl hydrolases were the dominant one and were found to be higher. If we see here, there are several diversity of glycosyl hydrolases identified. So we can conclude that this strain has a good potential to produce the lignocellulosic enzyme. So therefore, we have further developed this strain in a series of steps. So firstly, we have carried out the truncation of a transcriptional factor, which is the negative regulator for cellulases. We have truncated that Cre13. Then we have carried out protoplast fusion, diplodization, and then random mutagenesis of 2 round. And then we have knocked the another negative transcriptional factor, that is ACE1, and the developed strain which we got was GN11. So if we see here, the activities for GN11 strain was quite high as compared to the wild type strain, and the strain showed improved cellulase potential. So we tried to compare our strain with the DSM developed strain. So DSM is another biorefinery in US. They have also developed this strain Rasmussonia emersoni, and their last mutant, which they named, is TEC192, if we see in the table. So here we have tried to compare the secretome of our strain and their developed strain, and we found that the total glycosyl hydrolases and total cellulases, were higher in case of GN11 as indicated. And then we tried to identify that which are the dominating proteins which are found in the secretome of GN11, and we found endoglucanases, beta glucosidases, they are quite higher in case of the GN11. Therefore, we further screened those proteins, and we individually overexpressed those in Pichia pastoris. So if there are this is the list of the proteins which we have selected. And then we have individually overexpress them in the Pichia pastoris. So these include the cellulases, hemicellulases, that is beta xylosidases, then auxiliary activity enzyme, that is LPMOs. So we have purified them, characterized them, and studied their properties. But the goal behind this work was to develop the cocktail. So in the last stage, what we did, we took the crude cellulase from the developed strain, which I showed previously, GN11, and then we tried to boost the secretome of this strain with supplementing different monocomponent enzymes, which is shown over here in the list. So we supplemented those monocomponent to the crude cellulase, and then we screened those, and we found that the most potent one were the CBH, LPMO, and GH10. They showed the highest boosting capacity. So then we tried to mix them in different ratio. We optimized the ratio, and the final ratio which we got was this. And this cocktail was named as Ramazyme. But we have tried to evaluate our cocktail with the commercial cellulase. Like I mentioned, the goal was to study the competitiveness. So what we did, we used the equal protein loading rate. We supplemented our cocktail, and we used the substrate taken from Paraj Industries. So the substrate was steam and acid pre-treated rice straw, which we chose in our study. And then we tried to hydrolyze that substrate, and we found that the cocktail which we developed was quite competitive to that of the Selic CTEC 3. If we see, they are nearly closer, the saccharification efficacy. So in the last, I can conclude that the Rasmussonia emersoni strain, it has a high potential for production of cellulases. Secondly, the strain which we developed in our study can be equally effective and useful in the biorefineries, like they have developed, DSMS developed. So our strain can also be proved to be have good potential. And the third thing, we would like to conclude that the cocktail which we developed can also be the potent cocktail that can be used. So we have transferred one technology to GenCrest for enzyme production, and then we have filed the one patent. And currently our cocktail we have given to Paraj for their evaluation. Thank you.
I have just one question. What is the cost benefit ratio, right, in your case and the one with the industry?
Yes, actually we have not done the cost evaluation right now because we have sent our cocktails to the company. So once they evaluate our enzyme, then they will carry out the competitive cost. There's no approximation right now. I cannot say, it's inconclusive. Thank you.
Okay, so Dr. Raheja, it was really nice work.
Thank you.
And I hope that the potential cocktail you are developing or almost developed, I think so, because you have already transferred the technology to the Parag.
Yes, sir.
Okay. So have you like come across any kind of difficulty where in the cocktail, it's within the cocktail itself, the three enzymes or two enzymes, they are having antagonistic effect on each of them?
Yes, sir. While we were doing the optimization studies, so there were a list of the ratios which we used, and yes, there were some one that showed the antagonistic effect.
And second thing, because enzymes are highly specific to their substrate. So if you take the three enzyme in a cocktail, like enzyme E1, E2, and E3, and you are providing them the cellulose as a substrate. So how other enzymes actually targeting on the substrate cellulose, because they may be attacking on other kind of substrate?
Yes, sir. That is the problem which we also faced, but we have done the parallel strain development for another strain that are specific for those biomass, alkaline pre-treated biomass. We have separate enzyme and cocktails for that. For acid pre-treated, we have separate. So we are currently doing that.
Okay, okay. Thank you so
Thank
much.
you.
And I wish you all the best for your future.
Thank you.
And now the talk
One
is open.
question, one question.
Yes,
Yeah.
yes. Talk is open for the
Yes.
audience. Yeah,
Yes.
please.
Thank you. Thank you, sir. Okay. So my question here is like, how much FPU you are getting for this
Yeah, we got 4.5 FPU per gram.
per gram? And do you know how much FPU is IOCL is getting?
Yeah, it's quite high. It's 8 to 10 FPU.
It's 11.
Yeah, 8, 11.
I have worked 10 years in IOCL with the same technology.
Yes, ma'am.
So I just wanted to know about this thing.
Yes, ma'am.
Your cocktail. So how your cocktail is different from the Novozymes, because Novozymes is the pioneer for this, for the
Yes, ma'am.
cellulosic ethanol. So just what is the differentiation of your cocktail if we talk about the Novozymes or the IOCL cocktail?
Yeah, ma'am. If we talk about Novozymes, like they have not disclosed their composition, but still we know that they are using different fungus enzymes. They are using different sources for enzyme. But in our case, we have used Resmasonia emersoni, only the enzyme, and we have tried to boost the enzymes from its own secretome. So one thing we have in that.
So you are doing the molecular cloning and all these things?
Yes, yes, ma'am. We have individually expressed, like I mentioned, that monocomponents. Then we have tried to boost in its own secretome.
Okay, thank you so much.
Thank you, ma'am. Thank you. Thank you so much.
Okay, thank you. You should all give a huge round of applause to Dr. Raheja, and not only to her, and I think for all speakers, they deserve that because it is their hard work they are presenting here. So I would request all the audience, so when they finish their talk, please give them a huge round of applause as an appreciation. Thank you. Okay.
The next speaker is Dr. Neha Basotra. She's from Shahid Rajguru College of Applied Sciences for Women, University of Delhi. And her topic is scientific validation of ingenious Indian recipe bioactives against drug resistance pathogens from hospital wastewater.
Can you please channel? Yes.
So a very good afternoon to all of you. I am Dr. Neha Basotra, currently working as an assistant professor in the Department of Biochemistry, Shahid Rajguru College, Delhi University. So today I am going to present my work titled Exploring Spice-Based Antimicrobials to Combat Resistant Pathogens from Hospital Effluents, a One Health Perspective. To begin, I just want to say, I have a question, a very simple but a powerful question. What if the solution to antimicrobial resistance, it lies in our kitchens? So this is the agenda of my today's presentation. I'll begin with a short introduction, then problem statement, research gaps, the objectives of my study, a brief methodology, and the key findings. So before beginning, I just want to tell that this is just the preliminary study, the preliminary data that I am going to present here. So antimicrobial resistance, it's not a future threat now. Antimicrobial resistance is no more a future threat, but it's a present crisis. Multi-drug resistant microbes, they are increasing globally. And these hospital waste effluents, they are the main environmental reservoirs for these multi-drug resistant pathogens. Moreover, the conventional antibiotics efficacy is also declining. So there is an urgent need for an alternative, or you can say the adjunct antimicrobial strategy. So now the solution lies in our kitchen, that are the spices, the Indian spices. These Indian spices, they have the potential to act as an antimicrobial agents. Now question here is, why Indian spices? These Indian spices, these are rich in phenolics, terpenoids, and alkaloids, flavonoids, and other bioactive secondary metabolites. So they have documented antimicrobial properties in traditional medicine. These are consumed widely and culturally accepted, the usage of these spices. And more, however, one more important thing is that these spices, these are multi-target. If I talk about the conventional antibiotics, they can act on single targets, but these spices, Indian spices, they exhibit their antimicrobial action because they secrete a cocktail of active bioactive compounds such as these phenolics and terpenoids. Now, the problem here is most studies, they focus on the preparation of organic solvents for the extraction of these antimicrobial spices.
However, the domestic decoctions that we use are water-based, like the kadhas. These are water-based domestic decoctions. So this gap, that gap actually, what you can say, inspired me to carry out studies in water-based formulations of these spices. Now, there are basically 4 problems. First is the rise of synthetic drugs dependency and the side effects. Next is the global threat, or you can say the sudden emergence of these antimicrobial resistance. Next is the gap between the traditional wisdom and the scientific validation. And the last one is the underutilization of synergistic interactions in these spice blends. So the thing is, traditional wisdom exists. However, scientific validation of this traditional wisdom is missing. So this inspired us to carry out this study. And next, there are the key research gaps that we identified are the solvent bias, isolation versus synergism. That is, most studies, they focus on the isolation of a single compound from these spices. However, studies are there are scarcity of studies which actually focuses on the synergistic effects of the bioactive compounds that are present in these spices. Next is the lack of empirical validation for traditional decoctions, as I said. And lastly, limited research on multi-target antimicrobial action. As I said, these spices, they exhibit multi-target action. However, conventional antibiotics, the synthetic antibiotics, they can act only on single targets. So then, keeping all these things in mind, the following objectives were formulated. That is to isolate multi-drug resistant pathogens from samples collected from hospital effluent discharge points. Then to prepare aqueous extracts of selected Indian spices, to check antimicrobial efficacy and AMR analysis, then to study synergistic interactions between different spices, and to identify and characterize bioactive fractions of most potent spices, and lastly, to elucidate the molecular mechanisms underlying the antimicrobial effects of spice bioactives. As I said, this is just the preliminary study. Next is the methodology part. We collected the water samples from hospital effluent discharge points. And then these resistant pathogens, they were isolated using LB agar plates containing amoxicillin, azithromycin, and cefixime antibiotics. The spices that were used for this study were turmeric, clove, black pepper, bay leaf, cinnamon. And the antimicrobial evaluation of these spices that was carried out using agar well diffusion assay in Mueller-Hinton agar to check the zone of inhibition. Now, these are the key findings of my study. So, as you can see in this table, it shows that out of all the spices that were selected for this study, black pepper showed the maximum zone of inhibition, followed by clove and cinnamon. Moreover, synergistic studies were also carried out. However, the combination or the cocktails containing cinnamon and black pepper, they showed 17 millimeter of zone of inhibition, which is in antagonistic if I talk about the results of this black pepper. As black pepper, it shows the highest zone of inhibition, that is 29 millimeter. However, if I talk about clove and cinnamon, the 8 millimeter of zone of inhibition was observed. Now, this is the future framework of my study. I have done the preliminary work. Now, next, we are planning to go for the, to standardize the minimum inhibitory and bactericidal concentrations of these spices, and then to check the scientific, for the scientific validation of this traditional knowledge. We are also planning to carry out transcriptomic analysis in order to check the mechanistics, like how these spices are actually modifying the resistance in case of these resistant bacteria. And that's it. So this is the integrated future research framework. So to conclude, I would say that this study doesn't claim that these spices, Indian spices, can replace antibiotics. But yes, it strongly suggests that using the aqueous extracts of these spices, these can serve as potent antimicrobial adjuncts, and these can be used as environmentally sustainable alternatives. And these can also be used as resistance modifying agents. So perhaps the future of mitigating or combating these superbugs, it doesn't lie only in these synthetic laboratories, but it also lies on this scientific validation of this traditional knowledge. Thank you. I would be happy to take questions.
Thank you so much, Dr. Neha. Very interesting work you are doing.
But sir, this is just the preliminary work, so
Okay,
that's why I have
yes, yes,
results.
yes, yeah. We can understand because it is really it is very what you can say happy to see when the treatment is available in the home itself.
Yeah.
So why should we go
Right, sir.
somewhere else?
Right, sir.
So and all these our Indian spices, they have been very well documented in the Ayurveda and many other
Right.
system of medicines
sir.
for their remarkable bioactive properties. But at the same time, we also like looking forward actually. So all these basic work has already been done, I think, so
Yes,
to a
sir.
greater extent.
Yes, sir.
So what new thing, like what is the novelty you are considering for your work?
Sir, the major novelty is the aqueous formation. Like if you see the literature, most of these studies, they use this organic solvent-based extracts of these spices. However, in our study, we are focusing on aqueous extracts. The water-based decoctions, you can say, which are mimicking domestic decoctions. Like in Kada, we consume water-based decoctions of these spices.
Okay. Second thing, like when you say that the synthetic antibiotics, they are target oriented or target specific.
Yes, sir.
But now you are saying that this kind of traditional medicine, they are not target specific, rather they have
Yes.
broad specificity.
Yes, sir. Yes,
so
sir.
that you can cover like broad microbes
Yes,
under
sir.
that
Yes,
screen.
sir.
So which particular like aspect would be good for the future?
So
Whether
like,
target specific or the approach you are using that one.
sir, see, in target specific approach, basically we have seen there is an emergence of this antimicrobial resistance when we use these target based antibiotics. However, as these spices, these are multi-targeted. So what these are doing, these are actually modifying the resistance mechanism. So that would be more helpful. It's in my opinion, yeah.
Okay, okay. Thank you so much. Madam.
Yes, sir. Sorry, sir.
What different test organism did you use for testing the efficacy?
Sir, actually, we have checked the zone of inhibition using MHAGR. We just checked the zone of inhibition, and now further we will go for MIC. We will check the MIC and the MBC of these.
No, these clinical isolates you have taken. What?
No, sir, we have isolated these multi-drug resistant pathogens from hospital effluent waste sites. We actually isolated these pathogens in our lab, and then we checked the antimicrobial activity of these spices against these pathogens.
Okay.
Yes, sir.
If audience have any questions.
Ma'am, excellent work.
Thank you, ma'am.
So how many clinical isolates have you found in the effluents?
Ma'am, when we isolated these pathogens, in antibiotic containing plates, we got few colonies. However, in the plates which didn't contain any these antibiotics, there were so many colonies. I don't know the exact number, but in totality, I think 20 to 30 pathogens we isolated, and we have sent these for identification. We didn't identify these isolates yet.
Okay.
Yes, ma'am. Thank you.
Okay. So now we will be moving to the next speaker in the row. So now I invite Dr. Ajay Kumar Chauhan. He is from Sharda University, Noida, and his topic is biogenic conversion of graphite to graphene nanoparticles. Okay. Okay. So now, due to some unwanted reason, Dr. Ajay Kumar is not here. So we are moving to the next speaker. Next speaker is Shreya Biswas. She is from IIT Guwahati, and her topic is next generation plant-based galacto-oligosaccharides produced from potato galactan by Indo-beta-1,4 galactanase displays superior prebiotic selectivity and antioxidant properties. So I invite Dr. Shreya Biswas. Yeah.
This one for next, and this is David.
Good afternoon, Good afternoon, everyone. I'm Shreya. I'm working as a research scholar at IIT Guwahati under the supervision of Professor Arun Goel. So my presentation is on next generation plant-based galacto-oligosaccharides that has been produced from potato galactan by the activity of beta-1,4 endo galactase, which displays probiotic activity and antioxidant properties. So, as we all know, prebiotics are very important for gut health, microbiota, and its modulation. So, in nature and also commercially, there are so many probiotics that are available right now, and galacto-oligosaccharides are being one of them. But they are extracted from dairy, from lactose specifically. So, they are being dairy dependent. They are not very sustainable. So I have tried to search for a plant-based vegan galacto-oligosaccharides that can be sustainable and that has been underutilized till now. So the enzyme that I have used, and also we can see for the galacto-oligosaccharides that is present already in the commercial area, that has a glucose moiety at the end of the galacto-oligosaccharides, but the plant-based galacto-oligosaccharides that doesn't have any glucose. But because of the same structural feature, we are hypothesizing that the plant-based galacto-oligosaccharides will also give the same prebiotic and antioxidant activity. So these are the objectives that I have followed, that I have used first an enzyme from Acetovibrio thermosalum, that is a endo-beta-1,4 galactanase that is specifically works on beta-1,4 galactan. So I have taken potato galactan and I have used that enzyme to produce the galacto-oligosaccharides, and then I have tested the gastrointestinal stability and also its prebiotic activity and antioxidant activity. So here we can see that the enzyme ATGH53, that is from GH53 KZ family, that enzyme works on potato galactan and it produces galacto-oligosaccharides. Here I have taken a mixture of galacto-oligosaccharides of DP2 to DP5. And that we can see from the TLC and the HPLC analysis, that the highest ratio of the DP that is present in the mixture is of DP5 and the lowest is of DP2. And this galacto-oligosaccharides was the yield of the galacto-oligosaccharide that I got from mild conditions is 10% weight by weight concentration. Then for its stability in the gastrointestinal tract, I have checked in different conditions. First with the alpha amylase condition, and I have also taken inulin as a commercial prebiotic component to check for comparison. Here in all the cases for alpha amylase or simulated gastric juice or even simulated intestinal fluid, we can see the GOS has higher stability than inulin in all of the cases and all over different pH also. So we can tell that the GOS can be delivered successfully to the colon for further activity. Finally, I have checked with different prebiotic strains. So I have taken the GOS as a carbon source for growing different prebiotic or non-prebiotic bacteria. So I have taken some of the bifidobacterium bacteria, and I have also taken some lactobacillus strains for its growth. And here I have taken inulin as a positive control as a carbon source, and my GOS that I have used for the test. And also I have taken galactose as a minimal carbon source. And also I have taken one more sample that has no carbon source. Here, in all the cases, we can see that even though the galactose, that is the minimal carbon source, that has the highest OD in all the cases. But in Bifidobacterium, both inulin and both GOs has given similar activity. The growth rate in all the Bifidobacterium strains is almost similar for inulin and GOs. But in case of lactobacillus, the GOS is giving higher growth than inulin. Same, I have checked the fermentation behavior by checking the pH of the samples. And here we can see, even though in previous slide we have seen the galactose is giving higher growth rate, but the pH is dropping very rapidly, which is causing acidification. And that is not a suitable property for prebiotics. So here we can see in both inulin and both GOS and inulin, it is giving a mild drop in pH, which is very suitable for the colon environment. Further, I have checked the production of short chain fatty acids like acetate or propionate or butyrate, lactate, and formic acid also. And in all the cases, GOS fermentation produces enough amount of acetate, propionate, and also butyrate. And butyrate generation was very much notable in the Bifidobacterium species. Now, here in all the cases that we can see, there is a controlled and balanced release of short chain fatty acid, which is higher than inulin. So the short chain fatty acid profile also supports the colonic health and the gut homeostasis that is governed by GOS. After the prebiotic activity, I have also checked the antioxidant activity of the GOS. And here we have checked the antioxidant activity in a dose-dependent manner. So first, in the first picture that we can see the scavenging activity is increasing with dose dependent of GOS in both hydroxyl group and both DPPH. And also it is inhibiting the ascorbate oxidation, and the reducing activity is also governing by the concentration of GOS. So in conclusion, we can say that the mixture of GOS that is plant-based, that has superior gastrointestinal stability, and it also has a prebiotic activity, and it produces good gut microbiota, and it also shows a very good antioxidant activity with reference to its dose dependency. So in future prospect, we have plans for the in vivo and clinical validations, and also we are going to check its anti-cancer activity in further process. Thank you.
So Shreya, it is really very interesting and very significant work from both commercial and industrial point of view, I think. Yes. So as you have measured the antioxidant activity as well, why do you consider antioxidant activity as well as the other part of the work in combination?
Sir, I wanted to check the bioactivity for the substrate, for the component. So I was going to check on all the aspects: prebiotic activity, antioxidant activity, and also I have plans for the anti-cancer activity also, because that one component is giving me all of that activity.
Like we want to know that why only antioxidant activity, in what way it is going to be beneficial, or what would be the application of antioxidant activity in your case?
So
Finally,
if we're already using it as a prebiotic source, then when it is in the gut and all, the redox simulation and everything, it can be controlled by the GOS also. So it will be good for the health.
okay. So what kind of chemicals antioxidants are actually, and what is their role?
So, I have checked with DPPH scavenging and also
Yeah.
redox scavenging, and also the ascorbate inhibition, ascorbic
So
acid.
basically, they actually scavenge reactive oxygen species, ROS,
Yes.
and thereby they what you can say, increase the longevity of the cell cycle or cell life. Okay. So any other questions from the audience? Yeah.
I'll just continue with sir's question. What was the difference between all the three activities? Why did you test all the three? Why did you test all the three, like DPPH and all?
Ma'am, actually, I wanted to check with a dose-dependent manner. So I was checking for all the activity, ascorbic acid activity, DPPS scavenging, if it is giving the same manner, that dose dependency. Like with higher dose, it is showing higher activity. That is why I was checking for checking.
Yeah, my question is, what is the percentage of galactan in potato?
Sir, I have taken commercial potato galactan from magazine only.
Just for knowledge, how much of the galactan is present in potato?
Sir, I exactly don't know the percentage, but it is very high in potato.
That's not a good answer. Okay, anyway.
Actually, I have used the commercial
Yeah,
substrate
you
only.
use a commercial. Okay,
Yeah.
so the cost factor will be there. In your TLC slide that you have shown, the major oligosaccharide that you're getting by this activity is a biose.
No, sir. First two lanes are standard only.
Okay, they are
The
standard.
third lane is for my product.
Okay, so
It
which is the
depends.
major product
Yes,
you're getting? What is the major product you're getting?
sir, the major product is G5.
Okay. Okay, so how do how do you explain the growth of probiotics using GOS?
Sir, I have used it as a carbon source only.
Yeah. Now, what could be the basis for the probiotics to grow on in this? They must also be having the same activity
Uh,
because they are hydrolyzing it
yes, sir.
and then utilizing it. So, did you check that activity in the probiotics? That's my question. Thank you.
So if any questions from the audience? Okay. So there are no more questions. So thank you, Shreya. Now we'll be moving to the next speaker, and the next speaker is Dr. Ajay Kumar Chauhan. He is from Sharda University, and he will be delivering his talk on biogenic conversion of graphite to graphene nanoparticles using lignolytic enzymes from a halophilic isolate during solid state fermentation.
हाँ वही जस्ट वो सर एक्चुअली हमने खोली थी फिर हमने अनाउंस करा था तो शायद आप थे नहीं और
वही था जो आपने खोला ना अभी बायोजेनिक कन्वर्जन वाला just
ये नहीं है
ये नहीं इसके पहले भी ये भी नहीं है
हाँ ये नहीं इसमें देखिए खोल हाँ इसमें हाँ ये रे हो जाएगा स्लाइड हाँ हो
Okay, very good afternoon, distinguished professor and scientists and faculty members of Amity University and dear student. I'm Dr. Ajay Chauhan and is going to give my talk on biogenic conversion of graphite to graphene nanoparticle using lignolytic enzyme from halophilic isolate during solid state fermentation. So basically, first I want to tell you about what are the lignolytic enzyme. Basically, these are the enzyme which is produced by the white root fungus, and basically it is used for the degradation of lignin and complex substrate. So there are several enzymes which comes under the lignolytic enzymes. So the enzymes are lignin peroxidase, manganese peroxidase, lacase, versatile peroxidase, and dioxidizing peroxidase. The major application, as I told you, these enzymes are used for the degradation of complex molecule like synthetic dyes, lignin, and some of the complex antibiotics you can degrade using this enzyme. So as I have told you that I want to convert biogenic conversion of graphite into graphene oxide. So first I would like to tell you about why graphene oxide. So the graphene oxide, it is having the 200 times higher strength than the steel, and the density of this graphene oxide is similar to other carbon fiber. So if you can use graphene oxide as a nanomaterial, then it is going to be used in the different areas of research, like it can be used in solar cell, lithium ion industries, nuclear power plant, alcohol distillation, and several other applications are also there. So in the slide, you can clearly see that basically this is the solid state fermentation, where you can see this is the setup I have made during my solid state fermentation, where I am continuously supplying the sterile and moist air through the sperm. And this is the whole box is the humidifier, where I kept the 4 bottles continuously connected in a series. So I am continuously supplying the sterile air to the system, and this solid substrate is containing the bed of wheat bran, graphite, and the moist nutrient. And when I am continuously supplying this air, so my microbes are going and utilizing this oxygen, and after this utilizing this oxygen, they are producing their enzyme. And it is very difficult to monitor the growth of the system, but in the exist airflow, I have connected a CO2 sensor to monitor the growth of this halophilic isolate. So as the growth is increasing, the system is showing you the higher carbon dioxide content as a growth. And as the graphite as a material, why I have used the graphite as a material? Because the cost of the graphite is very lower. It is 500 kg, 500 rupees kg. And the important thing is that high thermal conductivity, because during the solid state fermentation, there is a challenge of mass and heat transfer. So my task was to reduce the means to increase the heat transfer rate. Heat transfer rate can be increased by increasing the thermal conductivity. During the system, I have monitored the carbon dioxide, and I have monitored the temperature as well, inside the flask and outside the flask as well. So due to this monitoring, I can clearly maintain and see that the temperature profile of this system was constant throughout the fermentation. As the other important parameter is the porosity of the graphite material, because the porosity of material is also important to retain some of the oxygen in the form of whatever the liquid water is present, and some of the oxygen get entrapped inside this porosity. So I have used this material. So while using this graphite as a material, you can clearly see that I have checked the several enzyme activity and compare the enzyme activity. And I have observed that when I have increased the graphite concentration during the solid state fermentation, the enzyme activity was significantly increased. And similar way, I had tried after post fermentation, I have tried to recover the graphite concentration as well. As I have increased the graphite concentration from 10% to 40%. Similarly, I have studied this study in the submerged fermentation as well, that my results are validated in the submerged fermentation as well as solid fermentation or not. So when I am increasing the graphite concentration beyond 40%, the recovery of this graphite material was very poor. So the 40% of graphite concentration was the optimized concentration for me. So the similar data was checked in the submerged fermentation. I have observed the significant increase of the lignolytic enzyme activity. The next is that I have checked the thermal conductivity of the material or the system, whether our hypothesis is working well or not. So I have checked the thermal conductivity of the system. So you can see that this is the conductivity of the material of the wheat bran, which is containing only the liquid culture. Next one is the graphite with the liquid culture. The thermal conductivity is around 1.05 Weber per meter per Kelvin. Then I have checked similar thing with the wheat bran and liquid and the culture. The the overall thermal conductivity was 0.45. Then I have checked the system where I am performing the solid state fermentation. So I can clearly see that the approximately 30% of the thermal conductivity of the system was increases. So as per our hypothesis, we want to increase the thermal conductivity and increase the heat transfer rate to remove the heat from the system. So during this process, we have observed certain changes on the surface of graphite. Why this activity is increasing? So what we did, we have taken the sample of graphite, recovered graphite, and I have done the sonication of that sample. So after the sonication sample, I have centrifuged the whole system with the different RPM, like 10,000, 2,000, 3,000, 4,000, 5,000, 6,000. So similar way, I have performed the repeated experiment, and I have observed that whatever the carbon present at the supernatant, it is suspended. The nanoparticle is suspended in the supernatant. So I have recovered the supernatant and collected on the surface of the filter paper. The nanoparticle is recovered, and I have just scan that liquid, which is liquid means supernatant, which is containing the graphite material, and scan that material. And I have observed the significant peak of 205 nanometer. There is a solder, and I have checked the literature and find out that this is something related to some of the particle get oxidized during the fermentation. So what I did next, so I have done the several characterization to check it that what if it is a graphite oxide or graphene oxide. So if graphite oxides are there, then there will be the monolayer, double layer structures are there. But if it is a multi-layer means multi-layer means it's a graphene oxide. So I have done the Raman data where I have got the peaks of ID, and this is the peak of ID, and this is the peak of ID, and this one the peak of the IG. And I have calculated the ratio of ID by IG, and I have found out that this is the nanoparticle which is related to graphene oxide. We have checked data with the FTIR for the first we have checked FTIR, then we have confirmed with the Raman, and after confirming with the Raman, we have done the XRD and find out that the tooth and theta angle which is relevant for the graphene oxide. So overall result from my study is that during the submerged fermentation, the enzyme production was significantly increases up to 23%. And when I have scaled it up in the packed bed type of reactor, I have made packed bed bioreactor for the solid state fermentation as well. And the enzyme activity increases up to 18 times during the solid state fermentation. And the important thing is that as we have targeted to increase the thermal conductivity, and thermal conductivity was significantly increased to 133%. And this data for the increasing thermal conductivity, right now, this company Fermentech lab, we have recently transferred this technology to them, and they are working on this area. So thank you so much. This is all from my presentation. Any question? This is the smart. So
Audience have any questions, please? The talk is open for the question right now.
okay.
Yeah, Dr. Ajay,
Yes, sir.
it's a nice work that I must say. But I didn't find what is the name of the microorganism that you're using?
It's a halophilic culture. Its name is Halopygeresvensis.
Okay.
It is extremophile.
But in your data, you didn't show any lignolytic activity. How can you directly relate it to lignolytic activity? There was also cellulase activity.
Yes, sir. It's a very interesting question from you, sir. Because there are the lignolytic enzyme, like the activity of lignolytic LIP is around 241 IU per gram.
Have you shown in any of the?
No, I have not shown here because the limitation.
Directly relates to lignolytic.
No. It's not directly related. Some of the data is still in publication. So there is one interesting thing is that the enzyme is showing some catalase positive result. And that catalase positive result is for the production of hydrogen peroxide. And this hydrogen peroxide is a co-substrate for manganese peroxide as well, and lignin peroxide peroxidase enzyme as well. So first, it is not directly, we can say that enzyme is directly converting substrate into product. So our system is producing hydrogen peroxide itself, and we have checked the hydrogen peroxide concentration as well. And that result is well versed for the production of this graphene nanoparticle.
Because what I feel is that you have to select few enzymes
Yes.
and then clone them and then try to print
Yes,
them
sir.
in
Yes, sir. Yes.
the mechanism. Thank you.
Yes.
One more thing, like as you told that the temperature was also increasing in the system.
Yes, sir.
Yeah, therefore you have conducted some thermoconductivity.
Yes, sir.
Yeah. So enzymes are also highly sensitive to the temperature. So have you checked the effect of temperature on the enzyme activity?
Yes, sir. As I told you, my microorganism is halophile. So whenever we are performing normal solid state fermentation, so temperature is significantly increases with the time. But as our culture is halophilic archaea, it is not clearly visible. So temperature profile I have already shown you. So the temperature profile is not significantly increasing during our fermentation process. I have kept sensor inside the solid state fermentation flask as well, and one sensor outside the flask as well. So our temperature profile is not increasing with the time. And our temperature, the key thing is that the enzyme is extracted from the halophilic archaea. So it has inherent property of working at higher temperature as well as alkaline with the saline, hypersaline condition as well.
Are you exploring any other approach beside the solid state fermentation?
Sir, presently I have hands-on with the submerged fermentation, like 20-liter fermentation facility. And right now I'm an assistant professor at Sarda University. So I have recently established the facility of 7-liter fermenter as well. So my student, they have isolated some of the bacterial and fungal strain as well. And some of the area I'm exploring because as I'm recently joined as a faculty. So some of the area I'm searching that how I can manage and do my research and to contribute in the scientific community.
Okay. Very good. Thank you. Thank you, Dr. Ajay. Yeah.
What probably may be the reason of the increase in packed bead reactor?
Can you repeat, ma'am, please?
What probably would have been the reason for the increase in the productivity in packed bead reactor?
Actually, ma'am, there is in the submerged fermentation that whenever you are adding any solid substrate, that solid substrate is in the submerged fermentation, that solid substrate is having the tendency to help in the agitation and increase the micro turbulence of the broth. So in the submerged fermentation, if I can add any compound, that can help in the increase in the activity. But in case of solid state fermentation, as I use graphite as a highly means conductive material, because whatever the heat is liberated, it should be transformed to the system of that particular. So that is why I can say that that can satisfy your answer, I guess.
What was the like level of graphite that has been converted to some like graphene you are telling?
Yes, sir. As I told you that 10% is the yield, 100 milligram I have taken as a used graphite and follicate with the sonication, and approximately 10 milligram of the graphene oxide I have recovered. So I can say that 10% yield was there.
Okay. Thank you, Dr. Ajay. Very nice work.
Yes.
And we say best of luck to you for your
Yes.
future endeavor. Thank you.
Yes, sir. Thank you so much, sir. Thank you.
So now we'll be moving to the next speaker, and our next speaker is Dr. Shalini Chaudhary. She is from our own MIT University, Noida, and her topic is isolation, screening, identification of coagulant protease produced. Okay. Yeah, she is pursuing PhD, but okay, we can still call her doctor, no problem, because ultimately she will be conferred doctorate very soon, hopefully. So let me once again invite Shalini Chaudhary. She is from Amity University, Noida. And her topic is isolation, screening, and identification of coagulant protease producing microbes for sustainable cheese manufacturing. So please, Salini, you can deliver your talk.
This is not the latest one. Okay, fine. It's fine.
Just a second.
रहने दीजिए क्या ये दिखा एक मिनट No, not this one. First one, which one is?
Just a second, मैं कर रहा हूँ दूसरी को.
यही है ना?
Yep. जी.
Okay. Good afternoon, respected judges and all the presentees here. My name is Shalini Chaudhary. I am doing PhD under the guidance of Dr. Smriti Srivastav from MIT University, Noida. First of all, I would like to thank this International Conference on Sustainable Biotechnology and Bioinnovation for providing me the opportunity to present my research work on the topic isolation, screening, and characterization of coagulant protease producing microbes for sustainable cheese manufacturing. Here, essentially, rennet, which is an animal protease, is protease used for traditional cheese production. But due to rising demand, dependency on animal source for production increases, and this leads to the ethical concern. Like animals were overexploited for production. So there is a huge need for finding the alternative and sustainable process to produce proteases for cheese production. So all the above, among the other alternatives, microbial proteases were found to be sustainable and suitable alternative for protease production. So as for industrial requirements, there are some challenges as high production capacity process parameters were
need to be more precise, and scaling up for commercial production were needed. So my research work is on basis on to overcome these gaps. Here is the workflow research, overall workflow of my research, where problems like I already mentioned were catered by using microbial proteases, which were collected and isolated from different sources and screened. After screened and quantitative analysis, we were targeted to found the potential protease producing microbes and their application in cheese production. So this is the work plan of my research, where samples were collected from different sites like slaughterhouse waste, dairy waste, which are good source of protein, and suitable sites to found the suitable potential protease producing microbes. From these microbes were isolated by different methods and followed by screening primarily on selective media. Then protease producing microbial isolates being produced in enriched media using skimmed milk as a substrate. After that, quantitative analysis were performed by enzyme assay and milk clotting assay. After that, comparative studies were done. These are the isolation and screening result. Here, graph 2 shows the pie chart where samples in which samples were collected from different sites, and the percentage shows the isolates we get from different sources, different sites. Figure 3 also represents the isolation of microbial isolates in agar plate method by agar plate method. After in figure 4, figure 4 shows screening result, primary screening result, where protease producing microbes were selected and studied on different pH. Their hydrolytic zones were studied. Here is the heat map, which shows the comparative study of primary screening on different pH, in which a neutral pH, which shows high and potential microbial isolates, and also on alkaline pH and acidic pH, they are also showing good results. But as for cheese production, after concluding the research literature, we continue go with the neutral pH. Here is the after that we do the milk clotting activity. We do the milk clotting assay and study the milk clotting activity of the selected isolates, according to which 6 isolates were found to have potential to used as a protease source in cheese production. Then in figure 7, the comparative studies have been done on protease activity, on the basis of protease activity, pH, and pH activity and coagulation activity of the isolates. This shows that strain 17N shows the highest and the potential protease activity and also the coagulant activity for cheese production. And this is the research outcome where potential protease producing microbial strains were isolated and being further analyzed on different pHs. A total of 288 microbial strains were screened and isolated. Among them, 280 were found to be positive on different pH. After quantitative analysis and comparative study, we found 17N isolate as a potential milk clotting enzyme having highest ratio of milk clotting activity to protease activity as 27.52. Here are the so we take 17 and isolate as for further research studies in which so there are the publication and the conferences which I have attended. So we are in the process of patenting on the coagulation efficiency. These are the references. Thank you.
Thank you, Salini. Very interesting piece of study. And now the talk is open for the questions from the audience.
What is the commercial source of microbial rennet?
Sir, in recently there are the fungal strains called Rhizomucor and Aspergillus were recently used in commercial production.
You're right.
Yes, sir.
But how your 17N is better than those?
Sir, actually, we we do the comparative study on ratio of milk clotting activity and the protease activity, which found to be suitable and potential for that actually. And the timing we at this point we are focusing on the timing, coagulation timing. So this result shows the maximum and the maximum potential by reducing the timing of coagulation.
Is it a gram positive or gram negative bacteria that you have isolated?
Sir, this is a gram positive.
Okay, so this protease enzymes, what is their speciality? Any specific characters they have?
So
or any protease enzymes can be used for the cheese making?
So no, sir. There should be a just like I am saying ratio which I am mentioning there. So for cheese production, there should be a minimum protease activity and the maximum coagulant activity. As if the protease activity is higher, then it affects the taste and texture of the cheese. So if the protease activity is high, then it can be a bitter in taste and.
Okay. So what substrate you have used for the enzyme activity measurement?
Sir, casein.
Casein.
Casein as a source, substrate.
Substrate. Okay.
Yes, sir.
Thank you. Okay.
In continuation with this, which he has asked, if the casein is the substrate, the production will be more of the protease, not of the coagulase.
Ma'am, for the enzyme activity, I have used casein as a substrate, but for the coagulation property, I have used skimmed milk for doing coagulation, for coagulation assay.
In both the cases, the casein is the compound which is responsible for the protease substrate.
Yes, ma'am.
In both, either you use skim milk or you use casein. Skim milk is the cheaper substrate, and casein is a little bit costlier side. So, but they are not for, they are for the coagulants, but the production is of the protease. But as per your requirement, protease should be less and coagulase should be high.
Protease activity, ma'am. Activity. तो मतलब I mean कि there are some proteases with high protease activity. There is the production of high wave during coagulation. So this 19 as just like I mentioned, तो इसमें जो 17S है उसमें curdling activity ज़्यादा है as compared to protease activity. So there is a protease activity, but it is less. They coagulate the protein more. That's why.
Okay.
One more question from Mr. Arjun.
Hello. Can you just tell me what are the key benefits of your study, like how you're going to implement in global future perspective and industrial scale?
Actually, we are studying for that actually. So at this point, we are just analyzing and screening the best isolate for cheese production. So for further, we are focusing on scaling and all the characterization for cheese production.
Okay, thank you.
Welcome.
So we should give her a huge round of applause. As I told you, not only to her, but all speakers deserves the huge round of applause, your appreciation. So now moving to the next speaker, our next speaker, 8 number, yeah, is Pranika Vasist. She is from Meerut Institute of Engineering and Technology, and she will discuss here assessment of wastewater discharge from multiple source in an educational institution, a case study of MIET campus. Okay. So uh, it seems that the Pranika is not here right now. So we have to move to the next speaker, and our next speaker is Gudhal Patanjali, and she is from the Amity University, Noida, and her topic is next generation nano-engineered essential oils, translating potent antifungal activity into field-ready control of Fusarium oxysporum. So if Godhal Patanjali is here, she may start her presentation.
So good afternoon. Good afternoon, respected chairpersons, distinguished delegates, and fellow researchers. Myself, Kudal Patanjali, research scholar from MIT Institute of Horticulture Studies and Research. Today, I will be presenting my work on next generation nano-engineered essential oils, translating potent antifungal activity into field-ready control of Fusarium oxysporum. So this work focuses on converting promising in vitro antifungal activity into practical field-level sustainable solutions. But before we talk about solutions, let us understand the magnitude of the problem we are dealing with. Fusarium wilt is one of the most destructive soil-borne disease in tomato. The pathogen colonizes the plant roots. It blocks the xylem vessel, disrupts water transport, which leads to severe wilting and yield loss. Its major problem is its long-term soil survival, which makes the management extremely difficult, especially in tropical regions. So when a pathogen can survive in soil for longer periods, the conventional strategies clearly become insufficient. So in that case, we need safer and smarter alternatives. So plant-derived essential oils are natural bioactive secondary metabolites. They show broad spectrum antifungal activity that work through multi-target mechanism, including membrane disruption and metabolic interference. Moreover, they are environmentally sustainable and residue-free. However, selecting the right essential oil requires scientific validation, which led us to our study rationale. So although essential oils are reported for their antifungal activity, their performance varies depending on their concentration, composition, and pathogen interaction. So therefore, before moving toward any formulation strategy, it was important to generate some comparative experimental data under controlled conditions. So this is the methodology. We have prepared 6 concentrations ranging from 500 to 3000 mg per liter. And we have performed in vitro antifungal screening using poison food technique. Also, dose response relationships were evaluated. So let us now look at the comparative results we have obtained from this screening. This slide provides the comparative antifungal, in vitro antifungal activity of all the 7 essential oil. As we can see from this bar chart that cinnamon bark oil demonstrated the highest antifungal efficacy even at lower doses. Orange oil also showed high efficacy, but it requires slightly higher concentration, and moringa oil showed the least inhibition even at higher concentration. So beyond comparing oils, it is important to check whether the activity follows a scientific dose response pattern. So as we can see here that all the oils exhibited clear dose dependent inhibition. As the concentration of the oil increases, inhibition also increases. And so we have generated this data in a heat map format to better visualize the intensity and sorry. So we have generated the data in a heat map format that shows that stronger oils reach complete inhibition faster. As we can see that cinnamon and orange oil reach the deep red zones rapidly, and moringa and mint remain in the lighter shades, showing the weak inhibition. So let us now look at the actual page, how this inhibition appears visually. So this slide provides the visual confirmation of decreased radial inhibition at increasing concentrations. We can clearly see the reduced colony diameter as the concentration of the oil increases. So we have identified the most effective oils, but strong lab results alone are not enough. So our next challenge is how to make them stable and effective at under field conditions. As we all know that essential oils are volatile and unstable under field conditions. So nano-encapsulation converts them into stabilized controlled release system, which enables prolonged persistence in soil and rhizosphere, and which reduces the application frequency, improves the cost efficiency, and it also enhances the farmer adaptability. But the main question is, how do these nano formulation act against the fungus? So this is the whole mechanism in which essential oil-based nanoparticles penetrate the plasma membrane, causing physical damage. Inside the cell, they induce the generation of reactive oxygen species, leading to oxidative stress. This disrupts DNA transcription and also downregulates ergosterol gene synthesis, impairing the membrane integrity. So this multi-target effect reduces the probability of resistance development. So based on all the experimental findings, cinnamon bark oil and orange oil showed highest antifungal activity among all the tested oils. So our next phase is to develop a botanical nano formulation which incorporate these oils either individually or in synergistic combination. So before I conclude, I would like to take a moment to acknowledge the guidance and support that made this study possible. I sincerely thank my supervisor, Dr. Abhishek Sharma, for his invaluable guidance and support throughout this research work. I also acknowledge Dr. Nutan Kaushik, head of the institute, for her leadership and encouragement of research excellence. And I'm extremely grateful for Amity Institute of Horticulture Studies and Research for providing this institutional support for this study. Thank you so much.
Thank you, Gudhal, for very nice work. And I think out of the box thinking behind encapsulation of the essential oil in the nanoparticle.
Thank you, sir.
Yeah. But there are few concern as well.
Yes, sir.
Essential oils and all the other kind of secondary metabolites, they are produced in extremely low quantity in the plant. Therefore, the large scale production of essential oil will remain a big challenge in your study actually. So second, and it will not be going to very cost effective as well.
Yes, sir.
So how will you cope up with these two problems?
Sir, actually, the initial cost would be high, but after nano encapsulation, the repeated spraying would be prevented as it reduces the application frequency. Also, the nano carrier, it will increase the surface area that will help in penetrating the fungal cell membrane very easily. Also, converts into control release system that will enable the slow release of the active ingredient.
Okay. Do you think that so many like chemical constituent present in the essential oil
Yes, sir.
may interfering with your nano formulation?
Sir, I don't think so, sir. So actually,
Why?
that particular ingredient is responsible for the antifungal activity. Like if we talk about cinnamon bark.
So why don't you go for that particular component for encapsulation instead of going for the whole essential oil?
So because essential oil doesn't contain a single compound, it contains so many compounds. Like if we, for example, cinnamon bark oil, it contains cinnamon aldehyde, which is the main component responsible for antifungal
So what is
activity.
the yield of the essential oil in that case, cinnamon?
So yield, as in if we take like the amount of oil, it will reduce during the nano encapsulation because it will prevent the repeated spraying.
Okay.
So the yield will be on.
Okay, I think Dr. Saab also have some questions
Yeah,
for you.
can you show me that slide again of the side, the dose dependent
Okay, sir.
effect?
Sir, this one?
Yeah. So what you did in this experiment? What is the percent inhibition? What do you mean by that?
So this dose dependent inhibition, it proves that the antifungal activity is concentration driven. It's not random.
Now, what is percent inhibition?
What is percentage inhibition?
On the y-axis, what is this percent inhibition that you're showing?
So that percentage inhibition, we have determined that using poison food technique. Like I will show you. Sorry. Sir, in this slide, we have screened the essential oil through poison food technique, and we have found the percentage inhibition of each oils. Like for orange, cinnamon bark, mint, eucalyptus, all the oils. So that percentage inhibition was higher for cinnamon bark and orange oil.
There's a reduction in the
Reduction in the colony diameter, sir, fungal colony diameter. Yes, sir.
Okay, and in the nanoparticle, what material are you going to use for making nanoparticles?
Sir, actually, as essential oils are volatile, so we will take some other carrier oil to make a composite oil phase. So
What's that?
So we can use castor oil, we can also use karanj oil, but we haven't decided that because that will be our next phase of our research.
That means we have not engineered anything as yet.
Sir, actually, we can take castor oil that is also approved under EPA. But sir, that is.
So these nanoparticles will be entering into the leaf?
Yes, sir, we will make the formulation. Then after, when we spray it, so it will penetrate the fungal cell membrane. So that nanoparticle will,
Fungal cell.
yeah, it will react with the fungal cell membrane more easily.
But what does, there's a fungal cell wall also is there?
Yes, sir.
You know what is a fungal cell wall is made up of?
So fungal cell is made of chitin.
Before coming to the membrane, it has to first encounter cell wall.
Yes, sir. Cell wall.
So how it is going to enter into the cell?
So the penetration will be much easier when it converts into
how it is going to dismember the chitin or cellulose which is present in the physics?
Actually, these studies, assessments that how they will work, it required more study and more.
One more thing. What is the size of the nanoparticles which you have prepared from the oil?
Sorry, ma'am, I don't understand.
It's a nano size. What is the size?
Ma'am, it's between 1 to 100 nanometer.
1 to 100 nanometer. So you have not checked yet?
Ma'am, it's in micron size now, but as I told you that it is our next phase to make a nano formulation. So that
it is not prepared till now.
Yeah, yes.
Not in a nano formulation at present.
Exactly, ma'am.
Yeah. And even like in your sir has rightly asked you that what was the percentage inhibition, then you told that it is zone of inhibition.
Yes,
Second thing, the concentration you are using there, I think that is also very high. It is going up to 3 mg per ml or something like that.
Yes, sir.
So with that high concentration, if you are trying to show the activity, so that may not be like very welcoming because people are already using micro concentration, microgram or nanogram concentration and demonstrating very significant antimicrobial activity. So you have to like think about that as well. Okay.
Yes.
Okay. Okay. So thank you so much. Good health.
Thank you
For
so
a very nice work.
Thank you so much.
And wish you all the best for your future work. Okay. Last है. Okay. Okay, so now we are moving to the next speaker, and she is Ashani Ray, and she is from National Institute of Technology, Durgapur. If she is present here, whether she is present or not. Okay. No, no. That is not possible. I don't think. We should not allow them on behalf of. Yes. So anyway, her topic was harnessing phenol-induced lignolytic enzymes for eco-friendly phenolic wastewater treatment. However, she is not present right now. So that's how we come to the end of this session. So all the speakers have completed their presentation. So now we will over to the Dr. Gurumurthy, session in charge for the proceeding further. Thank you.
This brings us to the conclusion of today's session on the applied environmental biotechnology and sustainability. And throughout the day, we have witnessed insightful discussions spanning applied biotechnology for sustainability, advanced bioprocessing for biofuels and bioproducts, and health and medicine. The presentations today have truly reflected innovations, scientific depth, and a shared commitment to a sustainable biotechnology and bioinnovations. With this, we would like to conclude this session. But before concluding, we would like to felicitate our esteemed chair sessions, because of which we can come through this end today. So before we conclude, I would like to invite Professor Debarati Paul, Professor Rajni Singh, to please come towards the stage and felicitate our session chairs. Felicitation team. May I invite session chair, Dr. Deepak Ganjewala, sir, please. Yes. Okay, the ladies first. Ladies first. I would like to invite Swati ma'am. And can I have a huge round of applause, please? Please, can you please applaud? Thank you, ma'am. The clapping should not be stopped, huh? Thank you.
On behalf of Amity University, Uttar Pradesh, Noida, and the organizing committee of International Conference on Sustainable Biotechnology and Bioinnovation, SBB 2026, we would like to express our sincere gratitude to our respected session chairs for their leadership and academic insight, to all our speakers for sharing their cutting-edge research and valuable time with us, to our faculty members, researchers, scholars, and all students for their enthusiastic participation and engaging discussion. Our heartful thanks also go to the organizing committee and our collaborating partners, the Biotech Research Society of India, International Bioprocessing Association, and the Center for Energy, Environment, and Sustainability for making this session a great success. We would also like to extend our gratitude to our gifting partners for their continued support. With this, we formally conclude the proceeding of day 2 in E2 Seminar Hall. Thank you so much for your presence.
A small announcement to make, please. Delegates and faculty, please move to the F1 lounge for F2 lounge, sorry, for the lunch. And the students, participants, please move to the F2 foyer for the lunch. Thank you. We are going to meet again after the lunch. Thank you once again. F1 lounge for the faculty and delegates.
University, Uttar Pradesh, in association with the Biotech Research Society of India, International Bioprocessing Association, and the Center for Energy, Environmental and Sustainability, with Unipic and IUth as a gifting partners, stands as a testament to our shared commitment towards sustainable biotechnology and bioinnovations. Today's program is designed to be both engaging and impactful, featuring insightful keynote lectures, thought-provoking presentations, and interactive sessions. We encourage all the presenters here to keep their talks clear, focused, and time-bound to ensure an enriching experience for everyone present here.
With that, let us begin today's proceedings. I now have the honor of introducing our session chairs, Dr. Vijay Kumar ji and Dr. Ayan Tarafdar ji. With that, our first session chair, Dr. Vijay Kumar ji, is presently working as an associate professor in the Institute of Neuropsychology and Neurosciences at Amity University, Noida. He has obtained his PhD in biophysics from All India Institute of Medical Sciences, New Delhi, and his research focused on the structural and biophysical characterization of heparin binding proteins of human seminal plasma through structural proteomics. He did his postdoctoral training from the University of Massachusetts Medical School, Massachusetts, USA. His postdoctoral research was focused on folding and aggregation studies of proteins involved in amyotrophic lateral sclerosis and frontotemporal dementia. His research interests focus on the area of protein biochemistry of neurodegenerative diseases, computational biophysics of protein folding, misfolding, and aggregation, and the role of toxic RNA species involved in the neurodegenerative diseases. He has published more than 80 publications in his field of specialization. He is a DAD alumni and was lauded with several awards like NESA Eminent Scientist in 2025 and Best Young Researcher by INSO in 2023. We welcome him and we would like to have a huge round of applause for our session chairs.
We request Dr.
Dr. Vijay
ji,
Kumar ji.
please take a lead and take a seat here
On the dais.
on the dais. Thank you so much, sir. Now
Can we have a round of applause, please, for our session chair?
Now I would like to introduce our next session chair, Dr. Ayan Tarafdar ji. Dr. Ayan is a scientist at ICAR Indian Veterinary Research Institute under the Ministry of Agriculture and Farmers Welfare, Government of India. His research area include food processing, waste management, biochar synthesis, and precision agriculture. He has over 170 scientific publications with 4,900 plus citations, and it holds 2 patents. And he is listed among the world's top 2% scientists. His work also integrates wastewater remediation and machine learning application in livestock farming. So I request you all to please give a big round of applause for him.
Thank you, sir. Thank you, sir. We now hand over the session to our session chairs so we can progress with the session, sir.
Thank you so much, sir.
Anyway, I can that's loud enough. So I welcome you all of you to in this second session of the day. So before we should start, let me introduce one thing sure that all the eminent speakers should stick on the time. They have been allotted for 10 minutes presentation, so 7 minutes talk and then 3-minute interactions. Okay.
Said the essence. Yes, sir.
Okay, so without any further delay, let me call the first speaker, Dr. Soumya Pandit from Sarda University. A huge round of applause.
Yes, sir.
So, am I audible? Good afternoon to all the dignitaries judges. Thank you for giving me the opportunity to present here. So today I am going to discuss a topic on bioelectricity energy recovery from dark fermentation effluent for powering low power microcontroller systems using a stacked microbial fuel cell. So we did this work at Sharda University Biopositive Lab. So let's start. Okay. Okay, so if you see the trend of the fuel, we started with coal basically, and then we moved from solid to liquid. We started with petroleum, diesel, we moved to petrol. Then we are using gasoline for aviation fuel, and we are using CNG for cars and our things and buses and all these things. And in future, we are going to use hydrogen and fuel cell. So the next era is for hydrogen and fuel cell. The demand is very high for these particular things. So we have to take into account of the pollution and the global warming. So in order to curb the menace of the global warming, we need something which will reduce the anthropogenic gases production. And for that, the option is hydrogen. So if you see the hydrogen production, the biohydrogen is particularly carbon neutral among all other hydrogen production. So what are the options that we have is dark fermentative biohydrogen production, direct biophotolysis, photofermentation, indirect biophotolysis. Apart from dark fermentation, all the others production system, they have some loopholes and the production kinetics is very, very less. So therefore, we have another option that is called microbial electrochemical system. So in this particular project, we have concluded these two and we try to find energy recovery using these systems. So what is this two-stage process? In two-stage process, first in dark fermentation, it produces theoretically 4 mole of hydrogen per mole of glucose. Okay, so rest amount of carbon with hydrogen product, that is the volatile fatty acid, we cannot throw it out. So what is the other option? The other option is we are going to use it either in microbial electrolysis cell or in microbial fuel cell for directly electricity generation. If it is microbial electrolysis cell, it would have been produced hydrogen production. So in this particular project, we are focusing on these things. So if you see what is the advantage of microbial fuel cell, we will be able to find that the microbial fuel cell can directly generate electricity using some microbes, which is called electroactive microbes. Like we used to give our electron to oxygen, which is the electron acceptor. These bacteria are by nature, they used to give their electron to the some metals or some oxidized metals or nitrate. But when we put an electrode, they used to donate their electrons to the electrode, and as a result, there is electricity production because of the potential difference, because there is a lot of negative charge on the surface of the electrode. But the problem with a single cell microbial fuel cell is that we can generate only 1.1 volt per cells. That is not enough to drive some good decentralized power system. So for that we need to go for stack system. That means in series connection, like this battery, or like a say a power battery or a motor. We can see an inverter. We can see there is a series connection of many batteries, and we can club it together. So based on that, we have this kind of research gap, that is a sluggish oxygen hydrogen production due to low pH, and because of volatile fatty acid, we cannot throw it out. So we need to utilize it for microbial fuel cell purpose. So we have this kind of gas collector system. You can see here that we can see here that there is a reactor in which we are producing hydrogen, and then the carbon dioxide we are putting in a trapper. Trapper contains KOH, okay, and KOH is absorbing the carbon dioxide. So only hydrogen is producing, and using the PV is equal to nRT, we can calculate what is the amount of hydrogen and what is the molarity of the hydrogen. The rest amount of volatile fatty acid which will be remain inside the reactor, that we will utilize for electricity production. So we are using ceramic reactor, which cost only 5 to 6 rupees, only 5 to 6 rupees per liter. So this is a really low cost ceramic fuel cell that we have used, and this is a one-time investment. So you can see here, we can generate at least up to 3 liter per liter of liquid, which is around 30 gram of COD that microbes can bear for biohydrogen production. With the time, because it's a batch reaction anaerobic, the pH is getting decreased, and volatile fatty acid like acetate and butyrate is producing a lot. So we try to utilize these things and fit it in a like contour plot, and using a surface plot, and we found that using Taguchi method, pH is the most important, and therefore we need to add sodium carbonate for this microbial fuel cell production. So this is the results of microbial fuel cell production. We can see that DF spent media, we can generate as much as 20 watt per meter cube. 20 watt per meter cube of electricity that is directly usable. Okay, and then we club it with electrical department, and they have developed a circuit which is DC-DC booster, and then they used MPPT technologies, and finally we use this for a continuous direct electricity, and we have run and power some 3 to 4 this LED bulb continuously for 4 to 5 hours. So if we see that calculation, we have done the calculation. This is a one-time investment. For entire reaction, we got the overall costing is around 8,000 rupees. But the production that we're getting, if we consider the current price of the hydrogen, it is much more than that. Also, the electricity that we are generating, that is a decentralized power system. That means that is a non-grid based power system. Anywhere we can fit it and we can utilize it, okay? Because we have to depend on this renewable energy. We cannot depend on non-renewable energy. Apart from solar, in future, this kind of biopower is going to play a very important role in future. So this is the overall conclusion. The biohydrogen production is around 2.8 to 3.2 liter per liter of liquid. The MFC stack was successfully using the volatile fatty acid rich in effluent. 20 watt per meter cube electricity we have generated during this MFC stack in series power connection. We have also reduced the voltage reversal problem in this fashion. The designed circuit helps in harvesting energy achieved during stack production. The proposed system can be utilized for decentralized power system. So this is some related papers that we have published in recent past, in last 2 years. Thank you. So I have 4 PhD students currently, and I'm thankful to the funding agencies, MSME, IHAB, and Artsure for funding this project. And that's it. I'm looking for some good collaborations. Thank you, sir.
Thank you. Thank you, Dr. Soumya. A wonderful presentation. The presentation is open for any questions from the audience, if there are any questions. Okay. So, Dr. Soumya, I had
Yes.
1 or 2 queries.
Yes.
In the initial, you mentioned that the traditional microbial fuel cells have 1.1 volts of electricity.
Yes,
Is that so? So if we were to translate your MFC, what is the typical voltage that you are getting? Because you have mentioned the power.
Sir, I'm getting around 900 millivolt because of there is some kind of internal resistance. The internal resistance is because the microbes by nature are organic in nature. Although they form a biofilm on the surface of the anode, and because of respiration, they are donating electrons and it's becoming negative. Because of the surface nature, because of the parental nature of the microbes, the charge transfer resistance is very high.
Okay.
So whenever we performed electrochemical analysis like Nyquist plot using electrochemical workstation, we found that there is significant amount of charge transfer resistance as well as diffusion resistance, which is called work resistance. So we try to reduce it, but this is not enough to power, say, for a small fan or for a mobile charger or for this thing. So we need to stack it. For example, in order to run this thing, say AC, we have a remote.
How many cells have you stacked?
Sir, in this particular case, we have used eight reactors.
8
reactors
reactors.
in series connection.
And whatever the biohydrogen production is there, what is the storage and transportation mechanism? Is it stable?
So there is a gap between hydrogen production, be it bio or whatever, and hydrogen storage. So currently, for example, Professor Owen Sebastov, their team, they are working hard. They are using metal hydrides for absorption of hydrogen and desorption of the hydrogen. Otherwise, what will happen? Hydrogen is a very small molecule. The entire volume of the hydrogen as a storage will be very big. So transport cost will be very high. And that is the problem that today people are using CNG, not hydrogen. Otherwise,
That's why I raised the question, because you are producing a lot of hydrogen.
Much larger.
Right.
Yes, sir.
Okay. Thank you.
Thank you. Thank you.
So the next presenter is Dr. Soumyajit Chandra. The next presentation is by Dr. Arpita Roy, and they'll be presenting on boosting biohydrogen production from pre-treated sugarcane bagasse dust via coupled dark fermentation and microbial electrolysis cell. So, Dr. Arpita, the stage is all yours. Please make the presentation within the given time limit.
Okay. Okay. Good afternoon to all. So I'm Dr. Arpita. I'm working as an assistant professor in Sharda University Department of Biotech. So basically, I'm going to discuss about the boosting of biohydrogen production from pre-treated sugarcane bagasse's dust via coupled dark fermentation and microbial electrolysis cell. This work has been conducted by Mr. Soumyajit Chandra, and he's our PhD scholar working along with me and Dr. Soumya Pandit. So coming to the work. नहीं चला ये चल जाए. Okay, I'll be fine.
Okay, so this work is in align with the United Nations Sustainable Development Goals, and it basically contains the more or less three sustainable goal. That is goal number 7, which states that it's an affordable and clean energy. 12, that is a responsible consumption and production, and 13, that is climate action. And it also promotes the basically the concept of circular bioeconomy by using the waste to generate the electricity and byproducts. So coming to the transition from green future to produce the hydrogen, it is one of the potential and having the future scope. So according to the market survey, it was estimated by different society or different reports that in 2030 it is being a USD $1 billion market, which will gives more potential. So here, when we are talking about the production of biohydrogen, the agricultural residues, that is sugarcane bagasse, is used as a substrate, and cow dung is used as an inoculum, which provides the source of the microbe for the production. So as the basic concept is using the two-stage process, which already Dr. Pandit explained that here we use the dark fermentation, and instead of microbial fuel cell, here we use the microbial electrolyte cell. The concept is same, that the volatile fatty acid will produce by using the glucose, it will convert it into the acetate, and then carbon dioxide and hydrogen, and carbon dioxide is captured by the KHO and hydrogen is released. So this dark fermentation step is one step, and another step includes the MEC, and this particular is having the 2-chamber, or I would say 3-chamber system. One is anodic chamber, and second is cathodic chamber, and in between it is separated by a proton electron membrane. So the process is showing us the waste valorization, use of low energy input, and as well as the sustainable process. So this strategy is already matching with the TRL level 4, that is technology readiness level 4. And when we talk about the entire process, so here's the when we taken the sugarcane bagasses from the local market, and then that was pre-treated, because without pre-treatment it was showing a little lesser, around 35% less output than the with the treated one. So that sugarcane bagasses was treated with the alkali and ultrasound pre-treatment method, and then the sugarcane bagasse dust was produced, and this dust was used as in substrate in the dark fermentation process, where your biohydrogen was producing, and the spent media is again utilized for the MES cell, where it is converting and producing the energy. So the integration of two stage are there, which will produce biohydrogen as well as improve the energy recovery. So this is the the setup. So from approximately 25 gram per liter of substrate gives us around the 3 liter per liter of biohydrogen, and there is an energy recovery of 11%. Then it was observed when only one type of microbial fuel cell, microbial electrolysis cell was used, the energy recovery is almost 8.2%, and different parameters are also verified that with time, the increase in the production of hydrogen also increases. So when we talk about the costing of the entire system of both the two stages, so in case of dark fermentation, overall the grass bioreactor, the substrate, the cost of input energy and other items, it will approximately comes around 5,100. And when we talk about the microbial electrolysis cell, the entire apparatus cost around the 800. So approximately 5,900 or 6,000 will be approx cost of that system, which is quite cheaper and for the production of the hydrogen. So the concluding remark from this particular study was that the agricultural residue, that is sugarcane bagasse, which is actually a kind of a waste and you are not using. So it is one of the chief source of material and can be utilized for the substrate for the generation of the biohydrogen, and the overall 82% of COD was reduced in the combined process. And when we are doing the single stage process, it will gives us around 11% of energy. But when we are combining the both the stages, it will gives an enhancement of the 19.2% of that particular energy. Thank you.
Thank you so much, Dr. Arpita.
Thank you.
And it's open for questions from the audience. Yes. Is there a
Ma'am, that was a really nice talk.
Thank
Ma'am,
you.
I have a question. Like when we pre-treat the sugar cane, then they will release the furfuryls also along with the sugars. Ma'am, how you're targeting them to reduce the furfuryls and the inhibitors so that they won't interfere with the growth of microbes?
Okay, so basically here we follow the alkali treatment plus ultrasonic waves. So both will reduce that thing, and it will be like later on also heat treatments are used. So that will inhibit those particular components, and we can use as a bagasse. Dust. Basically dust was used.
But ma'am, pre-treatment is basically used to release the captured sugar, which is captured by lignin and all. That will ultimately release the inhibitors, right? But ma'am, they can't literally reduce the release of inhibitors. How are you targeting this in your work?
I think in our case, it doesn't interfere with
Okay.
that.
Okay,
So
okay.
that's why we didn't face such kind of problem.
Okay, ma'am. Thank you, ma'am.
Any other questions? So you have used this ultrasonic pretreatment for sugarcane bagasse. Do you feel that it is scalable in the long run? Because ultrasound processing, if it is done in a bath, it's not very effective. But if it is done with a probe, it's effective but not scalable. So what is your opinion for that?
For that, sir, we are actually trying to optimize more. As we saw from single stage to when it is a two-stage process, there's little bit of increment in the energy store. So we are keep on optimizing the processes, like what process we can opt for and how we get the maximum amount of production. So first we use like alkali and the ultrasonic, but we are trying different other processes as well.
Okay. So if we don't use ultrasonic and just alkali, will that significantly impact your results?
It has not been yet done.
Okay. Thank you. Thank you so much.
Thank you so much.
The next presentation is by Ishan Moitra from Amity University. And the presentation title is Unlocking the Co-Cultivation Strategies of Diatoms with Microbes for Growth Optimization and High-Value Metabolite Production. Ishan, the stage is yours.
Am I audible?
Yes, go ahead, please.
So good afternoon, everyone present here. I, Ishan Moitra, currently pursuing MSc in Biotechnology from MIT Institute of Biotechnology under the guidance of Dr. Archana Tiwari. I am here to present on the topic unlocking co-cultivation strategies of diatoms with microbes for growth optimization and high-value metabolite production. The rationale behind this topic is what is the positive and negative effects of co-culture of diatom on bacteria and fungi. Controlled culture experiments always require prior microbial isolation. Diatom bacteria interactions are extensively studied as compared to fungal associations. Many studies relies on omics approaches over culture-dependent analysis. It means co-culture experiment has been studied in silico mode rather than experiment by culture experimentation. Baseline quantification of culturable microbes is essential prior to control interaction studies, which means Which are the bacteria or fungi are best suitable for interaction with diatoms. Systematic isolation is necessary before controlled interaction studies, and understanding cultural associates is essential for reproducible laboratory interaction studies. So let me introduce to the topic. Introduce. Diatoms are unicellular eukaryotic microalgae which are present everywhere, are the major contributors to oxygen production and carbon fixation. Diatoms naturally associate with diverse bacteria and fungal communities. The diatom microbe interaction affects growth, metabolism, and their biomass productivity. Microbial associations influence nutrient cycling. Co-cultivation strategies are being explored for biomass and metabolite enhancement, and their interactions is essentially biotechnological applications for sustainable development. So the next is diatom bacteria interaction. This is the illustration of how diatom bacteria interacts. Like diatom release organic carbon for bacterial growth. Bacteria supply vitamins B12 to diatoms. Inorganic nutrients regulate mutual growth response. Nutrient exchange enhances metabolic efficiency. These interactions promote growth modulation and ecosystem balance. Co-cultivation system improves biomass productivity compared to monocultures. Bacterial communities influence diatom lipid accumulation and metabolite production. So the next is diatom fungi interaction. Fungi associate with diatoms through saprophytic and parasitic interactions. Filamentous fungi aid in biomass harvesting through bioflocculation. In this, large clumps are formed, known as flocks. Fungal interactions helps in nutrient recycling. Chytride parasites diatoms by their penetration structures. Fungal diatom system influence bloom dynamics and energy efficiency, and have potential application in low energy biomass harvesting. So this is the experimental what we have did in our lab. The first part is isolation identification of microbes from wastewater. We took media, sterilized it, and used isolation technique, streaking and spreading, and it was incubated. After incubation, colonies were formed, and the colonies were counted by colony counter. To identify a microbe morphologically, we used staining and was examined under the microscope. The second part tells about is the workflow in which a media is sterilized, and the isolated microbe is cultured with diatom. So this is the result. This is the fungal colony plate, which has a count of 8.1 into 10 raised to power 2 CFU per ml. By the methyl blue staining, it showed filamentous shaped. For bacteria, colony count was 2.18 into 10 raised to power 3 CFU per ml. And staining was done using gram staining, which gave shape of comma, and it is a negative bacteria, gram negative. So I would like to conclude. We were able to establish baseline data for controlled co-culture experiment. Successful isolation of diatom associated microbes were achieved. Successfully cultured bacterial fungal isolates. Further studies will focus on growth kinetics, co-culture, and metabolite estimation, and understanding mechanism behind diatom microbe interaction. So these are the resources for my presentation, for my topic. Thank you.
Thank you, Ishan. Thank you. It was really a nice talk. Now, the stage is open for some questions.
Good afternoon, Ishan.
Good afternoon, ma'am.
You have given a very good presentation.
Thank you, ma'am.
And there are just 2 or 3 simple questions.
Okay.
What's the basic difference between an algae and a diatom?
Algae and
diatom.
diatoms are they are they are autotrophic eukaryotic.
No issues.
And
No issues. Secondly, in your flow chart, you have shown us the staining. So
Yes.
which stain you have used to stain the diatoms?
Wait, we haven't done that yet.
You haven't done yet, but which stain you are planning to use?
For diatoms, I think there is no staining.
There is no staining? Thank you.
Come to confirm, like, is it a diatom?
By the by microscopic analysis, there like microscopic SEM, with that how it looks like.
The SEM is clearly depict the presence of the diatoms in the media.
Can you repeat it? Sorry.
So SEM is good enough for the identification of the diatoms. SEM, you have just SEM, right?
Actually, no, yes.
Electron microscopy.
We can identify the diatom structure.
No issues, Ishan. Thank you.
Thank you.
Thank you, Ishan, for your insightful presentation, first of all.
Thank you.
It's just that I wanted to discuss that when we talk about the long-term stability of this co-cultivation systems.
Okay.
So how you are going to stabilize the metabolic system, or when we talk about this co-cultivation microbial approach, what you did through the diatoms and the bacterial approach. So how you're going to discuss this property, the metabolic stability, so that its property of mutualistic does not exceed to the competitive nature.
Yeah, yes, I understood it.
May I repeat?
No, no, it's
okay.
Yeah, so I know, but I am not able to recall it. I'm extremely sorry. I will get back to you next time.
Sure. Thank
Sorry.
you.
Okay. Okay, thank you. Thank you so much. Thank you.
Thank you, sir.
So the next presentation is by Dr. Pallavi Gupta from Amity University, and she'll be delivering a talk on bioprospecting microbial strains for biosurfactant production using waste cooking oil as a carbon source and exploring their multifaceted applications.
This one for next and this is laser.
Okay. Good afternoon, everyone. Myself, Pallavi Gupta. I'm pursuing my PhD in biotechnology from Amity University, Lucknow. So the title of my presentation is based on production of biosurfactant using waste cooking oil as a carbon source and to explore its multifaceted applications. So firstly, let me introduce that what are biosurfactants. As its name suggests that these are biologically synthesized surface active biomolecules, and these are extracellularly excreted by different microorganisms. These are the amphiphilic compounds, means they have both hydrophilic head and hydrophobic tail. So this dual nature of this molecule aligns with to interface to make among oil and water emulsion. So by reducing the surface and interfacial tension. Along with this, they can reduce they have very low CMC, means they are very highly effective at very low concentration. So what are the properties of the biosurfactant that makes it more advantageous over synthetic surfactants? As its name suggests that these are biologically derived, so they are easily biodegradable, these are very less toxic, these are eco-friendly, they are highly specific, and they are highly stable at different range of conditions like different pH, different saline conditions, different temperature. So all these properties of the biosurfactant makes it more advantageous over synthetic surfactants. So due to these properties, biosurfactant possess wide range of applications like in agriculture, in pharmaceuticals, in industries like in cosmetics, or in food industries. It can be used as a bioemulsifier. In environment, it can be used as a for bioremediation purpose. And it can be also used for heavy metal removal as well. So the aim of my study is to isolate biosurfactant producing isolates from the contaminated site using waste cooking oil as a sole carbon source, and to look at its multiple applications.
So here's the work summary of my research work, where I have collected the samples from waste dumping site, and the samples were enriched using waste cooking oil as a sole carbon source. Then the bacteria were isolated using serial dilution method, and the isolated strains were biochemically characterized to see the functional properties of the microorganisms. After biochemical characterization, then the screening of biosurfactant producing strains were done. There are several screening methods reported, like drop collapse assay, oil displacement, emulsification index, and surface tension. After screening, the potential strain was selected and was identified through 16S rRNA sequencing, and then the production and extraction was done, and the extracted biosurfactant was characterized through FTIR analysis, and then the multiple applications were tested of the isolated biosurfactants. So here are the results. I have collected the samples from the waste dumping site, which was located in Lucknow. And then the different bacteria were isolated, and based on the morphological differences, the bacterial strains were selected and purified and were assessed for screening assays. So depend upon the different screening assays, among 15 strains, the strain highlighted in red, like that is LS6, showed potential for screening of biosurfactant producing bacteria. So this strain was selected for further studies, and this strain was identified through 16S RNA sequencing as Citrobacter coceri, and is designated as Citrobacter coceri strain LS6. The flasks show the froth formation in biosurfactant culture, which indicates the surface active compound production. And the biosurfactant production using waste cooking oil gave the yield of about 2.8 grams per liter. Then the isolated biosurfactant was further characterized for its functional properties through FTIR analysis. As biosurfactant had different classes, so the biosurfactant produced from Citrobacter coceri was reported to be rhamnolipid type of biosurfactant. So we also tested the production kinetics and CMC of the biosurfactant. So for production kinetics, we saw that the OD of the bacterial growth increases up to 96 hours, along with the production yield, with the maximum yield of 2.8 grams per liter. And along with this, the surface tension reduces as the concentration of the biosurfactant increases. And the CMC of the biosurfactant was evaluated to be 60 mg per liter, which is very less, and the biosurfactant is highly effective. So we also tested the stability assessment of the isolated biosurfactant at different conditions like pH, salinity, and temperature. And the isolated biosurfactant showed high stability at all these parameters, suggesting its robustness and potential for different range of environmental conditions and its applications in different environmental fields. So after this, the applications of the biosurfactant was tested. For this, we have tested its emulsification of like different hydrocarbons, and it showed positive results for almost all of the different hydrocarbons. Along with this, we have also tested the crude oil removal efficiency of the isolated biosurfactant, where it reports that the isolated biosurfactant showed positive results as compared to control and the synthetic surfactants. Along with this, we also tested the antimicrobial activity against different pathogens, and we got positive results for this. Apart from this applications, we have also tested the assessment activity of biosurfactant in promoting seed germination, where we found that the biosurfactant at 0.5 gram per liter has a great potential had showed 100% germination within 5 days of incubation period without any fungal contamination. So through my study, I can conclude that the bacterial strains isolated from waste dumping site demonstrated potential for biosurfactant production, and out of 15 strains, one bacterial strain, namely Citrobacter coceri, was found to be potential using waste cooking oil as a carbon source, and the type of biosurfactant was rhamnolipid, and it shows wide range of applications in different fields. Thank you.
Thank you, Pallavi. It was really a very fast-paced talk. So I am like surprised that I could get some questions from the audience. Yes.
Hello, ma'am. That was a really nice talk. Actually, I have a question about the heterogeneity of waste cooking oil. Considering the heterogeneity and possible toxicity of waste cooking oil, how do you ensure consistency, scalability, and industrial viability of biosurfactant production?
Actually, with the waste cooking oil we collected is from we use it. It is a kind of like composition. It is made. It has the free fatty acids, oleic acid, palmitic acid, and leonic acid. We have collected it from the Amity University, matlab, canteen itself. So that was the waste cooking oil that we have collected from the samples, and we have used it as a carbon source.
Okay, thank you.
My simple question.
Yes, ma'am.
Your talk was very interesting and insightful. Where did you get this waste cooking oil?
I'm from the canteen.
Which canteen? Amity canteen.
I am from Lucknow campus.
Okay,
so
Lucknow
I have,
campus.
yes.
Okay, thank you.
Oh, yes, it was very nice talk given by you. I have just curiosity that the biosurfactant you produced or you isolated from the wildest strain, right?
Yeah.
So just want to take your note that how much it is feasible to go to the industrial scale for its production. Means a lot of studies are there where they are metabolically engineered microbes for overproduction, or we need actually overproduction of any biocompound for its scalability.
Yes.
So what is your note on that?
Yes, as we haven't tested on large scale production, but we can go for industrial applications. It is a pathogenic state, but we purify it. The biosurfactant it is a metabolic secondary metabolite. The biosurfactant is a kind of secondary metabolite which has been produced by the strain Citrobacter coceri. So we can produce it by giving up carbon source like waste cooking oil, and then we can go for a large scale production.
Okay, so this strain is pathogenic, right?
Yeah.
Okay, so is it means just a basic question that it is safe to go for a scale for an industrial approach?
Yeah, because we are not getting the cells. We are taking a cell-free supernatant for extraction.
You are not getting any kind of toxic compound in that extract. No,
we are
or you are
removing the cell,
purifying.
we are removing the cells. We are taking only the cell-free supernatant for extraction.
Okay, means you are doing purification of that one.
Purification.
Thank you.
I suppose there is no question. So, okay.
Thank you. Thank you, Pallavi. So one basic question, as you mentioned, you took the waste cooking oil from one canteen, but there could be difference in the composition of any other waste cooking oil from some another place.
Yes.
So will that impact your whole
Yes,
process?
it will impact on the production.
Hmm.
Like we have got the 2.8 grams per liter, but if it may depend upon the factors like upon the collection of the waste, the substrate.
So like you tried with other samples also, waste cooking oil samples?
Not yet, ma'am.
Not yet, only that one only. So have you done the composition analysis also for that?
No, ma'am, not yet.
Okay, all right. Thank you.
Dr. Pallavi, actually, my question is related to her.
Okay, sir.
You said that waste cooking oil is a supplement for carbon source, right? You are supplementing it
yes,
with the
sir.
carbon source. Have you analyzed the carbon content?
No, sir. We haven't analyzed
So
the carbon.
can we say that it's a supplement for carbon source?
Because sir, we haven't provided any other carbon source. We have just like we are growing the bacteria in a minimal media with providing carbon source WC waste cooking oil as a carbon source, and the production was seen over that. So that's why the waste cooking
But,
oil can
but
be
I
used.
think
as
you
a
should
carbon source.
analyze the carbon content.
Sure, sir. I'll
Other than-
analyze.
Thank you so much.
Thank you so much.
So next speaker is Aditi Anand from Amity University.
Aditya.
Why make it like this?
You can do anything.
Can we go from this?
Yeah. Just a second. This one for next, and this is for later. Yes. Yeah.
Good morning, everyone.
esteemed chairs and respected professors. My name is Aditi Anand, and I'm a BTech student from Amity University, Amity Institute of Biotechnology. Now, today I'm going to present my work on algal-based bioplastic and advancing packaging polymers towards a sustainable green economy. So we all know that plastics, like the consumption or the advancing plastics, are one of the major concerns that we have in our environment. And today my work actually shows how algae, which is one of the most sustainable resources present on Earth, can be utilized to make algal-based bioplastics. So as we all know, plastic have become an integral part of our life, and it is increasing rapidly. So globally, we actually produce 415 million tons of plastic every year, and this amount is like expected to increase twice by 2050. All of this plastic is actually mismanaged. It is not managed properly. Therefore, it degrades into microplastics and nanoplastic. This actually contaminates our soil, our rivers, our environment. They also come or get into our daily food cycles, and then also cause a lot of human diseases. Now, in order to tackle this, there is very much need in order to reduce the conventional plastic and shift towards a more biodegradable resources or bio-based plastics. Now, currently, most of the commercial bioplastic that is present or utilized are mainly derived from either starch or either cellulose crops. It's PLA or it's from PHA. Now, these bioplastics are actually take a large amount of agriculture resources or agricultural land in order to grow them. However, therefore, a lot of scientists are actually looking forward for anything that is non-food based resources so that they can also reduce the use of agricultural land. This is where algae comes in. Now, if I talk about algae is such a sustainable or such a wonderful resource. Like it utilizes CO2 while growing. It requires a small amount of land. It also can be grown on wastewater, which reduces our dependency on fresh water. It also grows rapidly as compared to the terrestrial plants. So they also release some polymeric substances and materials that can be used for bioplastic production. Now, there are many recent research that are going on algal-based bioplastics, and like they are being explored in various sectors, like in textile, there is textile sectors, then agriculture, biomedical uses. The algal-based bioplastic market is like small right now, but it's still growing at a rapid rate. Right now it's at 100 million, and it is expected to grow about 145 million by 2030 to 2034. Now, the main objective of my research work was to fabricate and reproduce a lab-scale agriculture bioplastic using simple materials and functional additives. And also, I did the characterization evaluation of the produced or the synthesized film. I evaluated the biodegradability, the water solubility, moisture content, and the chemical resistance. Now, this slide actually shows the methodology that I adopted in order to produce the particular film. I began with the biomass preparation. 10 gram of algal biomass was taken. I mixed it with 50 ml of distilled water, and I autoclaved it in order to extract the polymers. Then the homogenization was done. I homogenized it by mortar and pestle, and then taken it out from muslin cloth in order to get a uniform solvent. Now, later on, I also added some polymers. These are very necessary, like starch, glycerol. I added acetic acid, and then chitosan. Finally, the film casting was done. I poured it on a glass plate and dried it for 48 hours at 30 degrees Celsius. Now, further, I did the characterization, like biodegradability test. As I said before, the different characters are evaluated. I can talk about it in the future slides. Now, for the first characterization, I analyzed the color and moisture content, how the film looked. So the film that I produced actually had a little uneven surface. However, it had a uniform brown, green-brown color. It actually indicated algal biomass presence, and it had low impurities. No visible cracks. I didn't observe any visible cracks. There were no phase separations, and the film had a good structural integrity. Now, the moisture content was like 12.4% approximately. It showed a good moisture retention. Now, this moisture content is actually very important because it determines how much flexible the film can be, how much it can bend without breaking. And it is also very important for biodegradability because biodegradability depends on the microbial actions, and microbe needs water. So if a film has good moisture content, the microbe will act fast and the film will degrade pretty much easily. Now for the next observation, I did swelling percentage. I calculated the swelling percentage. I immersed the film in water, distilled water, for like 24 hours, and 76% swelling was observed. Film actually expanded, but it did not disintegrate. Now it actually showed it was stable, but water responsive structure. Now for the chemical resistant, I immersed the films in different chemicals. The first I immersed in 0.1 normal HCl. It actually reduced a little bit in volume. However, when I dissolved it in 0.1 normal NaOH and 50% ethanol, it showed a slight surface softening, but it remained intact. So it actually shows that in different chemicals, it can show a moderate resistance. The film can actually shows. Now the main test, which was the degradation test, I did the soil burial method. I uh, placed the film into the soil for like 60 days. And after 60 days, it had like approximately 62% biodegradability, which is actually very much impressive as compared to the conventional plastic films, which require like many years to degrade. The film showed decrease in weight, as is written here, 0.737 gram to 0.14 gram after retrieving it from the soil. Next, I did the water solubility test. Why I immersed in the water, and the film weight actually decreased from 0.32 gram to 0.23 gram for after 24 hours. Now, to summarize my entire presentation, I would like to say three main things, or three main points, or three main approaches. The first one is the feasibility and reproducibility. The film that I produced actually was based on water-based method. It is really easy to produce. I used like simple materials, like it was chitosan or it was starch, and these materials are like readily available in the market. Now, the next main important thing that I observed was the biodegradation. It had a huge biodegradation rate, like 62% is a very convincing biodegradation. And third, I would say for the future prospect, I am also looking forward to synthesizing and optimizing, like using this research as a baseline in order to optimize and produce a better film for packaging, and then evaluating the integrity or the mechanical strength or the tensile strength, and also the shelf life by like using different film, using that particular film in order to cover the food packaging or in food packaging sector. Thank you. I would like to thank my professors who actually helped me through the research.
Thank you, Dr.
No, sir, I'm a BTech student, Aditi.
Okay, thank you, Miss Aditi.
Thank you, sir.
It was a good presentation. I just have two quick questions. The first, you have added starch and chitosan and all. So all those materials already have film forming properties.
Yes, sir.
So have you taken them as any control film in which you have not used the algae?
Any control film? Have I synthesized any other film?
No, have you used starch and chitosan based film as a control in comparison with algae plus starch plus chitosan to see if that film, properties of the film is because of the algae or because of the starch or chitosan?
So starch and chitosan are actually provide the integrity, structural strength, mechanical strength. But I used algae in order to create a more biodegradable film. Although I did not do the comparison.
Madam, starch and chitosan are biodegradable.
Sir, I know.
So if you use algae, if it is improving any property of the film, you have to test it with the control. First point. Second point, you have also mentioned all these parameters, but for food packaging, the primary parameter is the oxygen and water vapor transmission rate.
Yes, sir.
The second parameter is it sea level? The sealability, if it is not there, it cannot be used for food package. So I think in your future studies, you should consider sealability and please add a control sample.
Yes, sir. Actually, I'm using this research as a baseline, and for in the future, I'll optimize the.
No, that's okay. But your baseline should start with control,
Okay,
not
sir.
with your
Okay, sir.
treatment.
Okay.
Okay. Thank you. Any other questions from?
Hello, Aditi. I have just one question. You have shown the biodegradability test, no?
Yes, sir.
Can you go back to that slide?
Yeah, sure, sir. Oh. Yeah.
You have shown this, how many days? You have 60 days, okay. You haven't mentioned the parameter of what is the depth you have used, and also the moisture content of the soil, because these two factor will be affecting the biodegradability of your film.
Actually, sir, I did it in the lab and I did it in the plastic cups that were available in my lab. And I did it like I took 7 centimeter of soil and placed it under 7 centimeter. And I like moisturized it daily in order to ensure
So
that.
have you measured the moisture content, how it is affected? Because moisture content will be affecting the biodegradability rate.
Sir, I did the normal moisture content as I shown here by oven driving, but I did not do it for the biodegradability test. I think you are asking me if I.
That is the film. I want to know whether biodegradability you have burying in the soil, na? The soil will be moisture. So
yes,
moisture will be affecting that one.
Yes.
Another suggestion that sir has told is if you are using for packaging, na, water vapor transmission rate you should study. It's very simple method. You can use it. Okay. Okay. Thank you. No more questions.
Thank you, sir.
Okay, thank you, Aditi. So the next presenter is Taniska Singhal from Guru Govind Singh Indraprasth University. The stage is now yours.
बैठा है ना टी-शर्ट उसको बोल थोड़ी देर आप माइक वाली कर लो This one? This one for next and this is later.
Okay, this one for next, right?
Yes.
Good afternoon, everyone. The topic for my presentation is genome editing of G protein coupled receptor and G protein G alpha subunit using CRISPR-Cas9 in rice cultivar Samba Masuri. I'm currently a MTech student in GGS IPU under the supervision of Raghuram sir. I have done this project. So, as we all know, rice is a staple food crop for more than half of the world's population. And improving rice for improving nitrogen absorption, nitrogen use efficiency, it is a very sustainable way for global food security. So, India ranks #1 in rice production according to USDA, and target nitrogen use efficiency is not solely controlled by the transporters or the or single genes. It is basically a signal. It is basically regulated by many signaling pathways. So one of which I have targeted is G protein signaling pathway. And rice basically has a NUE of less than 30%. It basically uses 30% of the applied nitrogen from the fertilizers. Two genes I have targeted, RGPCR and RGA1 gene. The objectives of my study were to basically construct the RGA1 and RGPCR in PRGB31 and cloning them into the E. coli. Standardizing the callus induction and regeneration process for rice variety Samba Masuri. Why Samba Masuri? Samba Masuri is basically a elite variety in India, and it is produced by most in our country, and it has a great yield. So targeting Samba Masuri would be sustainable for future research. And the third objective was to mobilize these positive construct into agrobacterium. So methodology involved preparation of E. coli competent cells, isolation of our vector, linearization followed by ligating guide RNA into the vector, transforming them into E. coli and confirming the transformation basically. Simultaneously, callus induction was also performed and callus regeneration was also performed. So these are basically the results. The vector size is 15 kb. So we were able to isolate vector successfully and linearization was done. We basically integrated the guide RNA into our vector, and it was for selection we have used 3 methods. First, LB agar with kanamycin as a antibiotic resistance, and the colonies were able to grow in LB agar. Next was confirmation using HPT primers, and lastly, it was confirmed by Sanger sequencing that the guide RNAs has successfully integrated into the vector. These positive constructs were successfully transformed into the agrobacterium, and it was selected on LB, kanamycin, and rifampicin antibiotic resistant plates. Callus induction was done for Samba Masuri. Rice was inoculated on callus induction media. And after 15 days, it was seeing the embryogenic callus efficiency was around 73%, which is a good percentage for transforming these calluses. And these calluses were grown on regeneration media with different kinds of hormones concentrations. The two are mentioned here. And after 10 days, the embryogenic calluses were shifted on to the shoot induction media. So the process was basically standardized for these rice variety. In conclusion, the guide RNA constructs targeting RGA1 gene and GPCR gene. These were successfully designed and cloned into PRGB31 CRISPR-Cas9 vector. Positive clones were confirmed through antibiotic selection, HPT confirmation, and Sanger sequencing. Efficient callus induction and regeneration protocol was optimized, achieving 73% embryogenic callus efficiency. And the future perspective involves the agrobacterium mediated transformation using the validated CRISPR constructs, molecular screening of these edited plants, and evaluation of edited lives for NUE, nitrogen use efficiency, growth, yield traits, abiotic stress tolerance, off-target analysis to ensure editing precision, and field level evaluation under reduced nitrogen input for sustainable agriculture. I would like to acknowledge my supervisor and SBB for giving me this platform. Thank you.
Thank you so much. So, any question? You people should ask question. Okay, so now the onus is mine only, okay? So the first question, how will you select the guide RNA?
How will I select the guide RNA? The guide RNA, basically, I've used ChopChop to create the guide RNAs. So the target gene was inserted, gene sequence was inserted in ChopChop, and it basically gives a list of guide RNAs. So I basically selected the starting exon so that the protein machinery could be affected, and the results could be like the knockouts could be created basically.
So do you think that one guide RNA can do their job for two different genes or two different proteins?
No.
So you have
guide
to
RNA
identify a specific guide RNA.
Yes.
How will you do this thing?
How will I? Guide RNAs were created for both the genes separately, basically. It was RGA1 had a separate guide RNA and RGPCR had a separate guide RNA. And what we are planning to do is to create a knockout separately first and then synergistically to see the effect.
So now RGA1 is a G alpha protein.
Yes.
If I am understanding correctly. Now suppose there are two different G alpha proteins. Okay. Let us suppose RGA1 and RGA2. How will you specifically design a guide RNA for RGA1 and RGA2? Specific.
Specific. So that I don't know actually.
So you have done all the CRISPR-Cas9 knockdown by your own.
Yes, sir.
That is commendable. Thank you.
Thank you, sir.
The next presentation is by Vishwanath Yadav on unlocking bamboo recalcitrance for bioethanol production through optimized delignification and tailor-made enzyme-based DSSF. What?
for the next 10 years.
So, hello. Good afternoon, everyone. Myself, Vishwanath Yadav. I am a PhD scholar at Indian Institute of Technology, Guwahati. Today, I am going to present my work on unlocking bamboo recalcitrance for bioethanol production through optimizing delignification of bamboo as well as integrated process of DSSF fermentation. So, bioethanol, as we all know, is the current most demanding product in the market because it is said that it is suited alternative of the fossil fuels. So currently more than 110 billion liter of ethanol is produced in globally, and 95% of which is currently made from the food items, generally corn and these things. So all these plant-based material consist of lignocellulosic biomass, which is composed of three major components: cellulose, hemicellulose, and lignin, which are complexly arranged with each other to form a cell wall of a plant. So why I choose bamboo for bioethanol production? The four major factor I choose bamboo were: it can be grown fast, it have high yield per hectare area, it requires very less input to grow, and for main important reason was the biochemical composition of bamboo, which was it has higher cellulosic content present. So whole cellulosic refinery can be divided into these three parts: pre-treatment, saccharification, and fermentation. Through integration of these three processes, final bioethanol is achieved. So what I have targeted is these research gaps which are the being a good cellulosic source. Bamboo has not been explored that much for ethanol production, as well as commercial cocktails present in the market for saccharification of lignocellulose are not that effective for every biomass. So we developed a tailor-made enzyme system from recombinant enzymes to produce for the saccharification of bamboo. And in last, we make an integrated process for fermentation of bamboo using Saccharomyces cerevisiae for production of higher ethanol yield. So proceeding with my first objective, which was effectively delignifying the bamboo biomass. So for that, we have used RSM CCD model for effective delignification and retention of total carbohydrate content in the bamboo. For that, we have chosen three independent variables, which were nitric acid concentration, pre-treatment time, and temperature of pre-treatment. And against these three variables, we calculated delignification percent and total carbohydrate content as the response. This was the model summary for my RSM CCD model, and sorry. These were the 20 experiments I performed in RSM CCD predicted model, and against them, each experiment response was mentioned. And further on statistical optimization, we got the optimized condition of 4.92% HNO3 with 132 degrees Celsius pre-treatment temperature and 150-minute pre-treatment time. Through upon validation of these pre-treatment condition, we achieved almost 90% delignification percent with 613 mg per gram total carbohydrate content in my solid stock after pre-treatment, which was closely aligned with the predicted value of the model, which was 95.6% delignification and 665 for mg per gram TCC. This was the comparative analysis, compositional analysis of raw and pre-treated bamboo for 100 gram. Further, these results were validated by structural analysis, which completely aligned with my study of stating that significant delignification was achieved along with the total carbohydrate content retention. So further, after pre-treatment optimization, we performed the saccharification with tailor-made recombinant enzyme cocktail, which consists of the cellulases and hemicellulases, which were cloned and expressed in our own lab in bacterial systems. And further, the optimization was performed for the cocktail composition using the de-optimal experiment design. And further on statistical analysis, we found the optimized ratio of these enzymes, which was 45.3, 28.4, 10, and 16 of these enzymes. And using this recombinant enzyme ratio, we performed the validation, and we found that 446 mg per gram of total reducing sugar was achieved after saccharification using this recombinant enzyme cocktail. Further, and validated cocktail showed the enzyme activity of 8.8 FPU per ml. Further, we performed thermal stability of this cocktail, and we found that after 72 hours of incubation at 35 degrees Celsius, the enzyme showed 64% retention of cellulase activity along with 68% of cellulase activity. And after successfully delignifying the bamboo and sacrificing it with recombinant enzyme cocktail, we proceeded with the fermentation process, in which we integrated saccharification and fermentation process through a DSSF process, which was delayed inoculation, simultaneous saccharification and fermentation. In this, we first sacrificed the pre-treated bamboo for 19 hours, and further, after that, we have added inoculum of Saccharomyces cerevisiae and performed the fermentation. So first, we have optimized the DSSF process by taking 3 independent variables by RSM-CCD model, which were pH time, biomass loading, and Triton X. Triton X is the surfactant which enhances the saccharification efficiency of the system. This was the model design, and these were the 24 experiments.
Vishwanath, please conclude.
Yes, sir. So after upon validation of this experiment, we found that 19.2 hour pH time, biomass loading was 85.8 gram per liter, and surfactant was 1.5 gram per liter. And with this, we achieved 20.5 gram per liter ethanol yield, which was higher than the predicted value of model, that was 18.6. And further, this validation was again performed in the bioreactor system with 2 liter reactor volume, and we achieved even higher 22.4 gram per liter ethanol titer with that. So to conclude, in this study, we have successfully performed optimize the pre-treatment of the bamboo, followed by the development of a recombinant bamboo-specific recombinant crude enzyme cocktail. And further, with integration of the saccharification and fermentation process, we have achieved a 22.4 gram per liter ethanol titer along with yield of 0.44 gram per gram. So finally, I would like to acknowledge my professor, PhD supervisor, Professor Arun Goyal, and Indian Institute of Technology, along with the funding agencies which have funded this project, along with my PhD stipends. And finally, I would like to thank this conference, SBB 2026, and MIT University for giving me this opportunity.
Thank you. Thank you for the wonderful presentation. Any questions from the house? No questions? So, okay, I'll be the one to ask. You have mentioned that your bamboo, you have done the pre-treatment studies?
Yes, sir.
So in that, have you found any inhibitors that are there for, that is the first question.
Yes.
Second, you are using both cellulase and xylanase together. So if you study the kinetics of fermentation, if you have done, have you considered any interaction effects that maybe one is degrading the other, or there is some competitive effect that is reducing the bioethanol yield?
Sorry, sir. Last part I did not get. I am using cellulases along with xylanases together.
Is there any competitive effect of using both the enzymes together?
cellulase and xylanase.
Yes.
Actually, sir, first we performed it with only cellulases. The efficiency was pretty less as compared to the combined cocktail, because the biomass which we are using is there's significant amount of hemicellulose present in the pre-treated biomass. So to completely access the cellulose, we need to remove the hemicellulose. So in my study, I have seen the synergistic effect between cellulases and hemicellulases, and they are eventually increasing the efficiency, saccharification efficiency.
Cellulases and xylanases.
Yes, sir.
Okay. So you have seen synergistic
Yes,
effect
sir.
between them. Is there a 1:1 ratio, or the ratio is?
No, sir. Ratio was actually for there was a chimeric enzyme of endoglucanase and beta glucosidase, which was 45.3%, and CPH was 28.4%, cellobiohydrolase. And xylanase and xylosidase were 10.2 and 16.1%.
Okay. And what do you think will be the economics of this process? Means considering that enzymatic saccharification could be expensive, and you are using two of them.
Yes, sir. So one part of this study, which I have not shown in this presentation, is we are trying to optimize the process for production of these proteins so that we can minimize the cost by high cell density and lowering the production cost. That's why we are not purifying this protein. We are using them in crude form so that cost can be reduced significantly. If we will go for purification of these enzymes, this will be really very costly.
Have you considered microbial processes for this ethanol production?
Not actually, sir. We have performed with, we have done literature review for everything, but we have carried forward with enzymatic. And for sir inhibitor question, we have screened through HPLC, we have screened for multiple inhibitors, but in this process, we could not find any. Generally, it is known that with HNO3, inhibitors are seen, but in my study, I could not find any. There could be any reason, but I did not find any. So we carried forward with this study.
Okay, thank you so much.
Sir, I have one question. Did you find any interference of dihydroxyfurfural during this production?
Yes, sir. That's what I'm asking. We did not find any inhibitor in solid fraction after pre-treatment. While washing, whatever inhibitors were there, they washed out.
Because Dr. Lignin is supposed to come and interfere with these things.
Sorry, you're not.
Lignin is supposed to come.
That is what I have done in pre-treatment. I have tried to significantly reduce the lignin content. I have achieved almost 90% of delignification. So already there was only 20 gram per liter. As you can see in this slide.
It's okay. I think you can discuss it over tea.
Yeah, sure.
Because we are running short on time. So I'll just invite. Thank you so much. I'll invite the next presenter, Milsi Pal, and their presentation topic is construction of shared soil stacked plant microbial fuel cell using nutrient film technology and hydroponics for bioelectricity production and its performance evaluation.
Thank you so much for giving me this opportunity. My name is Milsi and I am from Delhi Technological University. So today I am here to present my work on construction of shared soil stacked plant microbial fuel cell, and I integrated it with nutrient film technology, which is a kind of hydroponic setup. And also I have evaluated the performance of this setup as well. So let's dive into it. So first we will go through the motivation that why I did this work. As we all know that now global demand of renewable energy is increasing day by day, and we have set certain targets on national and global level as well. So I will discuss few out of them. global annual capacity of electricity generation for India in 2024 was 8,900 gigawatts. And it is estimated to produce 12,000 gigawatt by 2030. And we have target of at least using 70% of renewable energy out of it. So we can see from this data that there is huge demand of increasing renewable energy sources nowadays. If we talk about Indian annual capacity, so it was 440 gigawatt in 2023, and 43% of it we are using the renewable energy, and we have a global estimated target of 11,000 gigawatt till 2030, and we have 16% target of renewable energy for this as well. And if we talk about energy demand by 2040, it is estimated at 39 PWH. And if we talk about Indian energy, then it's 19 PWH. So I will discuss about plant microbial fuel cell a little bit. So what basically it is a biochemical system in which we integrate plants and the microbes to generate electricity. Okay. And this can be a source of clean and green energy, and it helps us achieving our role of zero CO2 emissions target at earlier levels. And yes, it can address a lot of problems like global warming, climate change, and there is also one term which is microbial fuel cell, which we were doing a lot of research earlier on microbial fuel cell, but we have now upgraded to plant microbial fuel cell, and it is 18 times more efficient than microbial fuel cell. There is a reason behind it, that is because in plant microbial fuel cell we are adding plants, so microbes can take plant exudates as their source to feed on this. So microbes can perform better and generate electricity. So how microbes generate electricity, we will see this on our latest slides. So let us now go towards the methodology. So first we will see the materials used. First I will tell about the plants which I use. So two plants I used, which one is Epomia purpurea, and common name for this plant is morning glory, and second one is Solanum lycopersicum, and this is very common. This is what we call tomato plant. So now let's discuss materials used. So I use seed for both the plants. So I started from the process of germination, and then net pots, as these are required in hydroponics technology and nutrient film technology systems, and then growth media that can support the roots and plant system, and then water pump to circulate the water through it, and an RO pipe to connect, and reservoir, titanium wire for the purpose of providing conductivity, and then carbon fiber cloth. This is for purpose of conductivity as well, and also this both titanium wire and carbon fiber cloth reduces the resistance of the system as well. And other than that, I used multimeter for measuring various readings such as current and voltage. And then nutrient media, because in hydroponics we require the nutrient media, so I used standard Hoagland media, which is majorly used in hydroponic systems. And other than that, NFT channels, because I integrated the system in nutrient film technology channels. So now let us discuss the design of plant microbial fuel cell, which we can see. So how I worked, I took net pot and then integrated cathode into the net pot, and then in the base of nutrient film technique channel, I implemented the anode in that portion. So there what happens is how nutrient film technique channel and plant microbial fuel cell works. There is a setup of which we can see in the working at the cathode. We can see at the top cathode, and in the lower portion we can see the anode. We can see this in working segment there. So in the anode section, there is a microbe biofilm, which mainly consists of anaerobic or facultative anaerobic bacteria, which are exoelectrogens or electrochemically active. So when they feed on plant substrate, they release electrons, and that's why we call those bacteria exoelectrogens or exoelectrogenic bacteria. So how plant microbial fuel cell works? So when we all know that plants synthesize their food by photosynthesis, and all the food that they synthesize cannot be assimilated. So rest of the unassimilated food is excreted through root exudates and okay. I will just conclude this in 1 minute. Okay. So,
You have 1 minute left.
okay. Okay, so I use Tughlakabad Biodiversity Park of a sewage discharge, which has highly facultative anaerobic bacteria. And let's come to results. These results are for Ipumia purpurea. I got R square value very close to 1. So this shows that results were accurate and what should be. And if I talk about the Solanum lycopersicum, then these are the results for lycopersicum as well. And I got parabolic values. And yeah, so I have also worked on polarization curves. And so what polarization curve shows? Polarization curve shows the significance in the real world of the plant microbial fuel cell, because here we compare plant density and current density on the scale of X and Y. So from the literature's review, we got 17 milliwatt per meter square of power, and we concluded 670. Yeah. Okay. So this is the comparison for both. I got more good results for the tomato, and maximum voltage for Ipumia purpurea was 762.3 millivolts, and for Solanum lycopersicum was 753.8. And if I talk about maximum power density, it was 614.41 for Ipumia purpurea and 670.13 for Solanum lycopersicum. Thank you so much for giving me this opportunity.
Thank you.
Yes.
Thank you, Ms. Milsey. Any
You're most
quick
welcome.
questions from the house? Any questions? Yeah.
So how you analyze the voltage and current?
Okay, so I used multimeter for that. Yeah. So we have titanium wires which was connected to our setups, and we connected multimeter to that, and I monitored and took the readings every day in a fixed time at 12 p.m. in a day. Yeah,
Thank you.
every day I took those
Any
readings.
reason behind 12 p.m.?
Yeah, because there is a sunlight, it is prone to show maximum reading at that time, because electrolytes can perform the better action. So I just took that time so that I can get uniform readings. Yeah.
Thank you.
Thank you so much.
You're most
So-
welcome. Yeah.
Thank you.
Thank you so much for providing.
Thank you, Milsey. So the next and the last presentation of this session is by Tasmia Falak. And the presentation topic is pyrolysis derived Tulda bamboo biochar for contaminant mitigation. Over to you, Tasmia.
Next is so.
So Tasmia, please try to wrap in 8 minutes only, okay?
Good evening, everyone. I am Tasmia Falak, and I am pursuing my PhD in environmental science from UPS Dehradun, and I am currently in second year. And today I am here to deliver my topic on pyrolysis-derived Tulda bamboo biochar for contaminant mitigation, which is carried out in collaboration with Korea University. So this is the outline of the project. This is the background of the slide. As we know that there is a multiple environmental problems coming across the global, from which the plastic pollution is one of the most global crisis that needs to be taken prior to mitigate that. So as we know that it's a very persistent pollutant, which is less than 5 millimeter, and it's a toxic pollutant. So there are basically main various bio-based materials present. Apart from that, biochar is one of the most cost-effective mitigation property that can be used for the mitigation of microplastic. So here, Tulda bamboo biomass has been selected, and for this, we will cover the SDG 6, 12, and 13. So before going to the study, this is one of the interaction mechanism which is carried out in when it is talked about biochar with respect to any contaminant. So there are various interaction mechanism that follows. First is electrostatic interaction, pore filling, intra-particle diffusion, hydrophobic interactions, hydrogen bonding, pi-pi interaction, all these factors that really have an impact on the mitigation. So this is the biochar which is selected from FRI Institute of Dehradun, which is a forestry-based biomass. That's a waste which is there in the campus. We selected and we have dried at 60 degree, shredded with this machine and powdered and made a biochar with a fixed bed pyrolysis reactor. We have optimized results for temperatures like 500, heating rate is 10 degrees centigrade per second, and residence time was 60 minutes. So this is the experimental study of pyrolysis of biomass, which is carried out in a fixed pyrolysis reactor. This is what a pyrolysis reactor looks like. And this is the chamber where 1 kg of biomass can be put into that. And the yield of the biochar have been shown here, 42 weight person, along with bio oil and syngas as a byproduct. I will show you one video that will show the experimental procedure that is followed. This is actually the furnace. This is the bio oil cylindrical gas where the bio oil catches, and the syngas is being produced here. So this is the specification mentioned here. What are the length and diameter and everything, it is mentioned. Due to the time limit, I cannot mention. So this is the characterization results of the biochar, which is here. Proximate analysis have been done, and we can see that moisture content have been reduced from 8.2 to 2.1, whereas fixed carbon analysis have been increased from 13 to 66.7. So here is the FTIR results has been shown here, which shows that compared to the biomass, the biochar have a very aromatic or functional properties present in them, whether it is OH groups or whether it is CH bonding, CO bonding. That is main properties which should be there in the material, whether it is bio-based or some that makes a part for the mitigation. This is the XRD analysis, which shows the amorphous nature of the material. This is the BT analysis of the biochar, where the surface properties have been increased from 0.69149 to 1.1399, which is shown here. And this is the comparative pore distribution table I have showed with respect to other biomass species such as sal leaves or peanut shell, which is not I have mentioned, but I have compared their pore diameter size. Here the Tulda biomass pore diameter is increases along 20 to 40 like angstrom, which is showing the mesoporous nature. So this is the biochar for mitigating, this is the application framework of biochar where can be utilized. So this biochar can be utilized for soil system. For example, this is the control condition we will take where soil plus microplastic will be there. And in the second, there will be microplastic plus soil plus biochar. So we will catch out the leachate of that water, and we will see that how much the leachate properties contains the microplastic or not. This is the novelty of the work that shows, that will be showing the effect, though the result is under the process. This experiment is actually carried out in Korea due to the low limitation of this. So this process is actually designed, and this will be carried out whether apart from biochar process that is done in India. So this is the conclusion part where we can say that Tulda biomass derived biochar was produced at 500 degree centigrade, showed a significant properties, whereas FTI XRD analysis showed. Future of the perspective which I can take out from this is that we can like modify this biochar with targeted, like we can make as an activated carbon, or we can modify the surface property to enhance its absorption capacity for different pollutants. And regeneration and reusability of what biochar can also be taken as one of the objective for the future perspective. So these are the references of the study. And on behalf of our team, we acknowledge all the Collaboration Hub program and the funding which is grant for this project. And Dean R&D and UPS fellowship for that and for this platform for giving this project to deliver this year.
Thank you so much, Ms. Tasmia. Wonderful work. Any questions from the audience? Yes. Mike, please.
So, Doctor, since you are preparing biochar under anaerobic conditions, like air is a very like nitrogen or gas that you are using.
Okay.
During pyrolysis, that is a very bad conductor of heat and electricity. So, did you think that what would be the cost if you can reduce the cost of the biochar using some low cost technology? Because that is very costly affair.
Yes, sir. Actually, first of all, I would like to say that this is free nitrogen-based biochar. That is not nitrogen was not supplied. And the second thing is this like cost effectiveness of biochar. I have went through, but this is very initial stage of my PhD work, and I will definitely carry out this point, and then we will forward in the coming year.
Thank you.
Really.
Any other questions? Any other questions? Okay, so I think that concludes our, thank you so much, Tasmia. So that concludes our session. So in this session, we had about 9 presentations. And I thank all the presenters for their wonderful talks. It was really interesting. We discussed about microbial fuel cells, biochar, saccharification, bioethanol, and everything else. I would also like to thank my chair, Dr. Vijay Kumar sir, for co-chairing with this session with me. And I thank the audience for listening so patiently and engaging with all the participants here. Thank you so much.
Before ending this session, we would like to felicitate our esteemed session chairs. So, sir, please don't leave. We'll continue now.
So may I request Dr. Sumedha Mohan ma'am and Dr. Tanu Elam ma'am to please facilitate our session chairs. May I request Dr. Vijay sir on the stage? Now may I request Dr. Aon? Thank you so much, sir. Thank you so much, ma'ams. Now, we would also like to extend our sincere thanks to our gifting partner for their continued support. With this, we formally conclude the proceeding of day 2 in E2 seminar. Thank you so much. अभी next session. अभी कर रहे हैं. Thank you so much, ma'am.
We are moving on to the third session.
नहीं रहना है.
Please do not leave anyone because we're starting with the final session of this day. We are starting with the final session of the day. And before that, we would like to invite Dr. Rajesh Kananji and Dr. Shailendra Shera ji to please be seated on the dais. Yes. Avinash, placards.
Sir, technical team को बोल दीजिए. बेटा.
Please do not leave. because we are continuing with the final session of the day. You have been a wonderful audience. We would like to continue with you all for the final session of the day. A huge round of applause for our audiences, please, first, because without you, our speakers, they cannot continue with their talk. So a huge round of applause for our audiences. Shall we start now? Okay. Now, good afternoon, everyone. On behalf of Amity University, Uttar Pradesh, Noida, and the organizing committee of the International Conference on Sustainable Biotechnology and Bioinnovations 2026, it gives us immense pleasure to welcome the last session of the day, Health and Medicine, and we extend a very warm welcome to our esteemed chairs, Dr. Shailendra Singh Shera ji and Dr. Rajesh Kannan ji.
Whose contribution to environmental biotechnology continued to inspire us in research and innovation. We also warmly acknowledge our session in charge, Dr. Guru Murthy ji, for his guidance and support in shaping this session. We would like to special thank to and welcome to our distinguished invited and plenary speakers, respected faculty members, researchers, industry experts, and our enthusiastic students joining us today. Your presence truly enriches the platform for knowledge, exchange, and collaborations. This conference, organized by Amity University, Uttar Pradesh, in association with the Biotech Research Society of India, International Bioprocessing Association, and the Center for Energy, Environment, and Sustainability, with Unipic and iYouth as a gifting partner, stands as a testament to our shared commitment towards sustainable biotechnology and bioinnovations. Today's program is designed to be both engaging and impactful, featuring insightful keynote lectures, thoughtful provoking presentations, and interactive discussions. We encourage all presenters to keep their talks clear, focused, and time-bound to ensure an enriching experience for everyone present here.
With that, let us begin today's proceedings. I now have the honor of introducing our first session chair, Dr. Rajesh Kannan ji. Dr. Rajesh Kannan ji, currently working as Professor and Head Biotechnology at Sharda University, Greater Noida, with 18 years of experience in teaching and research in nanobiotechnology and neurobiology. He was trained in David Geffen School of Medicine, University of California, Los Angeles, and EBI for his doctoral studies. He was a scientist E at the Center for Nanoscience and Nanotechnology and founding head of Center for Molecular and Nanomedical Sciences, Satyabhama Institute of Science and Technology. He is the recipient of prestigious grants and awards from EMBO and IAEO, FAO, Vienna Grant, and also COB, UK. During his research career from 2006 till date, he is the principal investigator of multiple funded projects from DBT, DST, ICMR, and Ministry of Earth Sciences in nanomedicine, neurotherapeutics, and marine bioactives. He is also working on the embryogenesis mechanism in zebrafish and crustacean models. His research focuses on neuroactive natural products, smart nanocarriers for BBB delivery, neurodegenerative CRISPR-Cas9 disease models, and antimicrobial resistance using zebrafish embryos as a developmental and biomedical model. He has publications in High Impact Journal. We welcome you, sir, and we can have a huge round of applause for our session chair.
Now, I would like to introduce our next session chair, Dr. Shailendra Singh Shera. He is a biotechnologist and bioengineering researcher with expertise in spanning biomaterials, bioprocess engineering, fermentation technology, and translational biotechnology. He holds a PhD degree in biotechnology from IIT Varanasi, along with an MTech in biotechnology from NIT Durgapur. Dr. Shera's research interest focus on the development of biopolymeric scaffold for drug delivery and tissue engineering, green synthesis of nanoparticles, microbial and enzymatic bioprocess, optimization using statistical and artificial neural network models, and emerging areas such as 3D bioprinting and organoid development. He has successfully led and contributed to funded research projects supported by state and institutional funding agencies. He has authored peer-reviewed research publications, book chapters with international publications, reflecting sustained scholarly contribution across biomaterial and bioprocess domains. The addition to academic research, he is also a co-founder of Urban Agrotech Farm Private Limited, a government-sponsored startup focused on a tissue culture-based cultivation and commercialization of medicinal mushrooms under RKVY Raftar scheme. His profile reflects a strong integration of scientific rigor, innovation, and industry-oriented biotechnology translation. Please give a big loud of applause for Dr. Shera.
We welcome you, sir, and we would like to start with the session. For the participants, you have 7 minutes for speaking, 3 minutes for question answer rounds, and a PPT should not exceed more than 12 slides. With that, good luck, all the best, and the session is in the progress now.
अच्छा कब तक चलेगा
Now I like to start the session and call our first speaker, Dr. Suruchi Chaudhary from Amity University, Gwalior. Dr. Suruchi Chaudhary from Amity University, Gwalior. The topic is, title is Biogenic Gold Nanoparticle Synthesized Using Fico religiosa. Dr. Suruchi? No. Okay, I think she is not there. Next speaker, I like to call Dr. Soumi Sadhu from Sarda University. Okay.
This is from your pets.
Good afternoon, everyone. Today I am here. I am Dr. Shomi Sadhu, working as an associate professor at the Department of Life Science, Sharda School of Bioscience and Technology at the Sharda University, Greater Noida. So today I'm here to present my work on the safety and efficacy of plant-based nanoparticle coated dressing for application on chronic wounds. Okay, it's changing, right? So yeah, so at the moment we're dealing with widespread antibiotic resistance, right? It's a serious concern because of the overuse as well as the abuse of profound antibiotics. We see that we can easily get these antibiotics over the counter, which is really a concerning matter. And we do not even complete the regime of 4 to 5 days as advised by the physician. That's why we see that. And again, the use and the misuse of these antibiotics in the agriculture sector, in apiculture, in livestock cultivation, we see that it leads to antibiotic resistance throughout. So there is an immediate need to address this through use of sustainable biotechnology and through green nano platforms. So these nano platforms, they can penetrate the physical barriers without causing too much discomfort. So, and why nanotechnology? Nowadays, everybody is talking about this new technology. So the reason for this behind is it not only employs plant materials to create the biogenic nanoparticle, but also the reason the physical properties of these nanoparticles is being the high density of the binding sites on their surfaces, which enables them to easily attach to the bacterial cell surfaces. And the increased surface area facilitates the rapid and the efficient penetration through the biofilms as well. So coming to the results of my work. So my work was basically creating an wound healing patch. Okay, so due to constraint of number of slides and all, I have reduced it and directly moved on to the results, wherein I am showing my synthesis and characterization of certain plant compounds which were used to produce certain nanoparticles. I will not reveal the names of these nanoparticles because the patent is still pending. For the rest of the presentation, I'll just go with C1 and C2. Okay, so here we see the synthesis of production of the C1 nanoparticles using hydrothermal and the C2 metal nanoparticles using the green synthesis method. Below, we can see the UV-vis absorption spectrum as well as the fluorescence emission spectrum. Along with that, we can see the nanoparticle tracking analysis of the C1 nanoparticle, wherein we can see that the size is 38 and 44. However, the SEM image shows that there is a higher size due to accumulation during the dry powder formation. That is why the size has increased. Now coming to the antibacterial activities of these nanoparticles, as we can see that it has very good antimicrobial activity against both the gram-positive as well as the gram-negative bacteria, and it is almost equivalent to the positive control using streptomycin. Going to antifungal activities, we can see that its zone of inhibition is almost equivalent to the antibiotic, that is amphotericin B, both in Candida as well as in Aspergillus. So now the second, the slide C is showing the minimum inhibitory concentration in Staphylococcus, and D is showing the minimum inhibitory concentration in Pseudomonas. The E graph is showing the biofilm inhibition assay. Coming to the biocompatibility studies of the C1 and C2 nanoparticles, we can see that both of these nanoparticles have shown good biocompatibility, less of hemolysis, as well as less cytotoxicity in terms of cell viability. So now finally, we moved on to the preparation of this wound healing patch. Now for that, we used an electrospinning machine for that, in which we used PCl as well as PEO. This PCl stands for polycaprolactone, which is hydrophobic in nature, and therefore it has been used on the outside. And this PEO, on the other hand, this is polyethylene oxide, and it is having hydrophilic properties, which means that when it is used over the wound site, it can absorb the excess pus and that is released from the wound site. And these are the different concentrations in which the PCl, PEO was used. Along with that, we had also used banana nanofibers. This nanofibers was created from the pseudostem of banana and converted into dried and converted into nanofiber form, and then it was mixed in different concentrations in the patch. These are the preparations of the different patches in different concentrations. Out of that, we went forward with the testing of the final patch, which was patch 6, in which we found good antimicrobial activity. So this is the characterization of the patch. This is the FESM image showing this PEO with the C1 compound and the PCl with the banana nanofiber in the group. On your right-hand side, you can see this. Okay. Now, finally, coming to the in vivo evaluation of this wound dressings in the Wistar rat models. As we can see, this PC stands for the positive control, wherein a well-known antibiotic, that is mupirocin, was used. Then coming to the negative control, where the wound was kept open. Then coming to the vehicle control, where the patch was used having no components in it. Then coming to C1, and the combination was of C1 and C2. in which it was found that the C1 was showing the most most profound wound closing capacity. As demonstrated by the histopathological studies, we can see that in the test group 1 we are talking about, here we can see that there is good growth of the collagen remodeling, good angiogenesis, and faster tissue regeneration compared to the negative control, where we can see that there is too much of inflammatory cells that have infiltrated, and therefore the nature of the cells is not that good. This is the final evaluation of the wound contraction, in which we can see that in compound C1, it is showing 91% wound closure compared to the positive control, which is 71%, and without the drug, it is 80%. This is the biodegradability studies of the patch, in which we can see that almost all of the patches which were created by us by us were showing good biodegradability after a period of 40 days. And this, the one in pink over here, is the known patch that we use. That is, it's a band-aid. After 40 days, it is has shown no biodegradability at all, as we already know. So this is the graph showing the biodegradability of the patches. And in conclusion, we can say that these plant-based nanoparticle-coated dressings showed promising therapeutic potential in the rat model. Now it is we have to upskill and upgrade this work to human volunteers. So we have already applied for that project in DST. So let's see what happens. We can finally test in human models and see how effective is this patch. And it is the dressing not only exhibited desirable properties such as sustainability, biodegradability, and biocompatibility, but also showed good wound closure capacity. So I'd like to thank my lab members. This is Dr. Shankar, my collaborator, and my students. From the top, Ms. Swati, Mr. Shoaib, Ms. Bhargavi, and my lab. So, and also thank to Sharda University for providing me the funding. I got a seed funding for the completing this project, and to Amity University for providing me this platform. Thank you so much.
Hello. Any question from Soumya, participants? Do you like to ask any question? No. Dr. Soumya, I have some questions.
So it's show me. Dr. Soumya is someone else.
Oh, Dr. Soumi, sorry. Dr. Soumi, I have few questions. One, I want to know, you have done some soil biodegradability.
Yes, sir.
Have you tried this patch with simulated environment? Like, have you used protease, simulated body fluid?
No, not yet, sir.
Because for soil biodegradability, you have shown that it is degradable in nature.
Yes, yes, because both we were concerned only with the polymer, because rest of the
another thing is that you are trying to use it for the chronic wound environment, no?
Yes, yes.
So there is a
we have to keep on changing. It's not that you're saying that maybe it will degrade on our skin surface itself. That's what you're suggesting.
Yes, actually, the chronic wound environment is like protease environment is there. Okay,
yes,
so you
yes,
should include that in your study, because in research paper, they ask.
Okay,
Because
okay.
soil is something else, unless and until you prove that I am generating something like biodegradable thing. Okay, if you are using it in the chronic, they ask for these data. Have you done it in the simulated body fluid? Have you done it using the protease? Because wound environment itself is a very, very harsh
Yes,
environment.
that is true. But we have done the studies in vivo rat model, in which every 4 days we had changed the patch. We were using So it was a total 16 days evaluation study in the Vista rat model.
Another thing I cannot see one, since you are using on the wound. So where have you done the water vapor transmission rate study for?
No, we have not done that yet. We are actually, all those things are in pipeline because, there's issue of funds. So we have
No,
applied for larger funds.
WVTR you can do very easy. If you have a desiccator,
Mhm.
you can fill it in the small vials, cut the patch and drop it, put in the desiccator and see the loss of water vapor by simple. You can search it on the net. You
Yes,
can
sure.
get it.
Thank you so much for the
Can you
suggestions.
can you bring it to that model? Where you have shown the inhibition? Yes. This I want to ask. You have shown some in vivo studies. What is the design? How much Vista rat you have used, and have you done how many male, how many female?
It was mostly, as far as I remember, because this was outsourced. All of the
At least you know the
all the
design,
rats
na?
were male, because otherwise, if females and males cannot be kept in a single cage, it. So and then the vehicle, when each group had 5 rats.
Five rats.
Yes, 5 rats in each group, along with the positive as well as the negative control, along with the vehicle control, as well as
Because
two catchers.
nowadays the data they are asking is that diversity of the data. You have done more study only on the male. Okay, some papers ask
Yes,
okay,
again, I
how
was-
you are going to diversify the data.
Yes. So I am expecting in the next few months a larger fund in order to do this work. So that's why I cannot reveal my components, because if obviously things come to the fore, so I'll be upscaling this work. We're doing it in more number of models, as well as testing all the patches, all of the different formula patches, because we could only test patch 6. Maybe other variations in the components that would be used may show better data. We don't know that.
And
Because
another
it
one
could
more
not be
question,
tested.
one last question. I will not take a lot of time. Do I want to ask?
Of course, sir.
One question is that you are using nanoparticle. So can you tell me the nature, whether it's metallic or the organic nanoparticle?
One was organic in nature and one was metallic in nature.
metallic
But we
nan-
found, as I told you, this patch 6, it doesn't have, this is having only the C1, having only the organic nanoparticle, which was showing the best results. When it was combined with the metal one, that is C2, it wasn't showing as good
So
results.
why you are
Maybe.
using the nanoparticle in your patch?
Because that is the one having the antibacterial, antifungal activities. Yeah, that I have told itself.
That's one more question. That's
Yeah.
why I have asked. Have you seen the how this nanoparticle is getting released from your patch, like drug release study
Yes,
or nanoparticle release?
say is also being done, but the
Means
data
you
is
have
there, but I have not put it over here due to constraint. The data is there. There,
You
the
have
drugs
done?
are, yes, that has been done, definitely. Because
Thank
we have to put that in the paper.
Okay, thank you, Dr. Somi.
Thank you so much.
Dr. Swami, one question. What was the monodispersity or polydispersity nature? You have done DLS as well, right? What was the tentative size, percentage of monodispersity?
I do not remember that, so I'm sorry.
No, I think you have kept it. I think you have DLS you have done, right? DLS studies.
DLS, yes, it has been done.
You would have got some range, maximum range. What was its range? Like, size range.
About the nanoparticle
Yes,
size you're
yeah.
asking, yeah, it was between 38 and 44 nanometers range.
Okay.
And but as I told you, the same image was showing 500 nanometers.
That is
So
because of the
because of that-
clumsiness. Yeah, that is different.
because
Yeah.
you have to definitely take it in powder form, and whenever, because it's an organic nanoparticle, as I told you, its tendency is to agglomerate. Now I have actually put it, I've repeated the data, I've sent the sample to AIMS. Let's see what I get. I'm expecting that it will be better.
Have you determined the IC50 value before patching into the material?
Yes,
polymer?
that has been done. That's MIC value has been done. Then only the percentages
What
were
was-
decided.
what was it?
MIC value was around 1.56 mg per
No,
ml.
I'm not asking the microbial. I'm asking the quantification of the particle. Inhibitory IC50 concentration.
IC50 the IC50, I think it has been done because we did MTT assay. So
What is it? What is the range?
in MTT assay, we did IC50 for the biocompatibility assays. We have tested in PBMCs, and we found that it is almost even at 1 mg per ml, like 1000 microgram per ml. It is showing good biocompatibility. That is, the cells are still 70% viable.
Because since you are going to patch it over with the material, right?
Yes.
So if it is extremely high or extremely low, we do not know.
We could not.
So because you need to fix it, right? The amount of quantified material which you are going to.
It's been done in the percentages I showed, how much percentages has been used in each batch that has been shown, and that percentage has been determined from the MIC value as well as the IC50 value in compatibility studies.
Which amount showed better activity? Which range? Do you have any idea about that? Chris?
The range was between compatible, compared to the streptomycin, the positive control, we can see that it is showing at very high concentrations, that is 50 microgram per ml. But since 50 microgram per ml, if I come down to the MIC, it was at around 3 point something.
If it is 50 microgram per ml, if it is metallic particles, it might be calm.
That is why it was toxic in the when we were used it. But the organic one was not showing any kind of hemolysis or any kind of compatibility issues, so it was better. And the wound closure was also better because it's sort of a traditional medicine that has been used for long. We have taken from that.
Okay.
So.
Okay, thank you.
Thank you, sir.
Thank you, Soumi. Now I like to call the next speaker, Kaustubh Adhikari from Sri Venkateswara College of Engineering. Kaustubh Adhikari, no? 24, okay, thank you. Next, moving on to next speaker, Saurabh Singh Salaria from Dr. Bhimrao Ambedkar National Institute of Technology. Huh? Okay.
Next and this is for later. Next and this is for next 2 years.
Good evening, everyone. My topic. Yes. My topic is cellulose-based hydrogels with enhanced antimicrobial activity for wound healing applications. So starting with the introduction, hydrogels are 3D structures which are made from the hydrophilic polymers, which have higher capacity to retain and absorb fluids. The synthesis methodology involves sol gel methodology. Initially, a solution of polymer is prepared and then it is gelated using either physical or chemical treatment. Now, these are some of the advantages of using hydrogels as wound dressings, as they provide moist environment. They can absorb excessive wound exudate, and they can be used for delivering drugs and bioactive compounds, which can help in inducing anti-inflammatory, antibacterial, as well as antioxidative properties. These hydrogels made up from the natural polymers are biocompatible, and as compared to the traditional wound healing patches, they do not adhere to the surface of the wound, and they can also act as a extracellular matrix, and they can provide cellular support and regeneration for wound healing. This is the extraction methodology which we have used for cotton and kapok based fibers. Initially, they were treated using NaOH and followed by sodium hypochlorite and our organic acid to synthesize the smaller particles of the cellulose. Then those cellulose particles were sonicated to synthesize the cellulose nanofibers. And similarly, the sol gel methodology was used. Initially, the fibers were freeze thawed, then the cross-linker was added to form the cellulose-based hydrogen. This is the FTR analysis, which clearly shows the aliphatic groups as well as the glycosidic bonding, the COC as well as CH at 1160 and 2900. These are the same images. The image on the left shows the porous structure of the hydrogel, and the image on the right shows the CNF, the cellulose nanofiber structure. These were the loading and release studies which were done on the hydrogels. This is the antimicrobial property which was acquired by the hydrogel after the loading of the drug. As we can see, these plates were tested against both Staphylococcus aureus and E. coli, and the drug-loaded hydrogels showed high antimicrobial activity against the both E. coli and staph. Thank you.
Thank you, Saurabh. Now, I like participants to ask question. Any participants? Do you have any questions?
Yes. Yes, ma'am. Actually, we are trying to incorporate two type of drugs. One is the synthetic drug, which is well known established drug, and the second one we are trying to incorporate a natural compound. Natural compound. No, sir. Actually, ma'am, we are in the process of doing that, and the drug which we are using is still under process, and we have. Yes, ma'am. Yes, ma'am. Yes. Thank you, ma'am.
So some of the bacteria that produces capsules.
Yes, sir.
How do you think that how your things will be efficacious towards
You are
the
you are
capsules?
asking about the spore forming bacteria, sir?
Yes.
Okay. Actually, the synthetic drugs, sir, which we are incorporating in our hydrogel, it's known. It has a known antimicrobial activity against even drug-resistant bacteria also. That's why we have selected that. And for the natural compound, we are still on working to find that natural compound.
Whenever we used to
Yes.
whenever we used to study these things, we mostly use suspended growth of the microbes, but mostly we should carry out it in against biofilm. So biofilm is a very robust kind of
Yes.
things. Okay. So do you think that your materials will be efficacious towards biofilm?
Sir, you are asking about the
Microbial
biofilm formation, biofilm formation on the wound. That's you're asking about. Sir, same answer. The drug is quite potent against most of the microbes, as we have tested it against both gram-positive and gram-negative bacteria, as well as it has shown antimicrobial activity against several drug-resistant bacteria also. That's why, sir, we are using
Is there any chance that it can be a quorum quenching molecule?
quenching.
Quorum quenching molecule, like
Quenching in terms of chemistry, I've heard.
Thank you, Saurabh. Yes, sir. Thank you, Saurabh. It was an insightful presentation. I just have two questions. One is, how did you isolated the cellulose?
Yeah. Ma'am, we have pre-treated the cellulose using NaOH hypochlorite and removed the lignin part from it. Actually, I've used three sources. Two were plant-based sources, and the third one was bacterial. There was limitations of slides. That's why I haven't shown the bacterial one.
Okay,
Yes.
okay. And secondly, as you have shown that you have characterized the cellulose by using the SEM and the FTIR. So in FTIR, which peak that you talk about for the characterization?
Yes, ma'am.
Functional group?
Ma'am, for the presence of cellulose is essentially made up of the glucose units, and those are linked by glycosidic bonding. And ma'am, can I go to the slide?
Yeah.
Yes, ma'am. Here we can see COC at 1160. I've compared it with the literature, and this probably shows the glycosidic bonding in cellulose nanofibers, as well as the CH at 2900 aliphatic group.
Okay. And according with the SEM, your previous slide of the SEM,
Yes, ma'am.
yeah, this slide. So, but just looking with this SEM that you can characterize or you can identify that, yes, it's
No,
a
no, no, ma'am.
cellulose.
Actually, it is not for the ma'am characterization. It was just to make sure that we have synthesized cellulose nanofibers. They are in a nano range. We can further go for TEM to finalize. Yes.
Characterized. Thank you so much, sir.
Thank you.
Thank you.
So in your image, it shows the range of 1 micrometer, right?
Yes, sir.
So what might be the tentative pore size? Pore size.
Sir, these fiber these, sir,
Because you are talking about the pores, right?
yes,
Pores.
from your side, the
You have not
image,
measured it.
actually, we haven't calculated it, but these pore sizes actually, sir, the image on which you can see on your left is cellulose nanofibers. These fibers are essentially dissolved in the NaOH.
I know that because there are some spore-like structure, you know.
4.
So it is sputtered, right? Gold sputtered. Is it so? What's the technique used for coating this polymer?
Sir, we are using, I think, gold plated particles, gold plated.
Because your range is 1 micrometer. That's what I'm asking.
Yes, sir.
So
Sir, actually, we have got other images also.
can you show that?
Sir, I haven't bought my laptop in here. So, sir, those images are also showing these structures, but
Okay, it's fine.
sketches में हम नहीं हाँ जी.
But what do you mean that aerogel? What is that?
Sir, these are dried hydrogels. We cannot directly use the hydrogels for SEM imaging. उनको dry करना पड़ता है samples को. We have to dry them first.
Saurabh, can you bring me to that drug release study part?
This
yes.
one?
Yes, back. In figure that one, I am seeing that burst release, why it is happening?
Yes, sir. It shows the burst release in initial hours. Actually, we can further go for the release after 15 minutes. I've shown the release after hours, 1 hour, 2nd hour, 3rd hour, 4th hour.
Why it is so that it is?
Sir, these are just initial trials. We are still to optimize our studies. Actually, we are trying to functionalize the CNF for better absorption of the drug as well as better release at different pH.
You are in which year of PhD?
Sir, I am in 2nd year.
Okay, so you should know why it is doing. Bring to next slide.
Yes, sir.
See, you are a PhD scholar. That's why I want to ask you. You have shown the zone of inhibition here, but you haven't mentioned the size. Unless and until you mention the size in the slide, how we will be knowing that this out of this AB slide, which
Yes,
concentration is working?
Yeah.
Have you measured or not the diameter of the inhibition?
Yes, sir. You are absolutely right, sir. We will further integrate this into our studies. We are still to do MIC and MBC.
No, but whenever you show us anywhere, not here also, anywhere, you should mention plate also. You should also mention the what is the diameter of the zone of inhibition. Unless and until you mention it, the data is incomplete. Okay.
Yes.
These two suggestions I want to give you. Okay. Thank you, sir.
Thank you, sir.
Now I like to invite next speaker, Divya Kaushik from Sarda University.
Good evening, everyone present here. I am Divya Kaushik, PhD scholar from Department of Biotechnology, Sharda University, and I'll be presenting my work on the topic screening of beta-lactamase inhibitors from marine spawn symbiotic actinomycetes, SRK001. Antimicrobial resistance, as we all know, it is one of the most serious global health threat. And according to WHO Global Antibiotic Resistance Surveillance Report 2025, 1 in 6 bacterial infections are resistant to antimicrobial treatment. And we are witnessing rapid spread of multi-drug resistant pathogens. Frontline antibiotics are losing their effectiveness, and there is very limited pipeline for antibiotic development. Need for novel therapeutic interventions it is needed. So this crisis is particularly alarming in case of beta-lactam antibiotics, as beta-lactam antibiotics are the most widely prescribed class of antibiotics. So here in this image, we can see the resistance mechanisms adopted by bacteria. For example, efflux pumps, antibiotic altering enzymes, antibiotic degrading enzymes. And among these mechanisms, enzymatic degradation of antibiotics is one of the most clinically significant mechanism. And in case of beta-lactam antibiotics, this mechanism is through beta-lactamase enzyme. And if we look into the mechanism of action of beta-lactamase, it cleaves the beta-lactam ring of the beta-lactam antibiotics, and particularly it cleaves the amide bond. For example, in this diagram, we have shown penicillin. The highlighted part is beta-lactam ring of penicillin. And when the beta-lactamase acts on it, it cleaves the amide bond and it is converted into penicilloic acid. And it is actually the inactivated form of penicillin. This led us to explore marine environment as specifically marine sponges, because marine sponges are considered as microbial reservoirs as they contain 40 to 60% of microbial biomass. Marine environment are nutrient limited, they're highly competitive and chemically dynamic. Actinomycetes living in symbiotic relationship in marine sponges, they produce structurally diverse and novel secondary metabolites as part of their defense mechanisms. So coming to my objectives are isolation of beta-lactamase inhibitor producing actinomycetes from marine sponge, media optimization and production of beta-lactamase inhibitor, and purification of beta-lactamase inhibitor using TLC and RPHPLC techniques. So my methodology includes, first we collected the samples from south coastal regions of Tamil Nadu, particularly Kanyakumari. Then we isolated the marine actinomycetes and we performed molecular identification. Then we also did morphological characterization of actinomycetes. Later on, we did fermentation and extraction, and we confirmed the inhibition by beta-lactamase inhibition assay. Finally, we did separation of positive beta-lactamase inhibitor using TLC and RPHPLC techniques. So these are the results. First, we did molecular identification by 16S rRNA sequencing, and we saw a very high similarity with Streptomyces strain. It showed 98% similarity. And as we can see in the phylogenetic tree, that our strain SRK001, it is very closely related to Streptomyces species. Then we did morphological characterization. And a very distinct feature of actinomycetes is that the front image, color of the actinomycetes is different and the back color of actinomycetes is different. It is also showing pigmentation and metabolite exudation. And after that, we did the beta-lactamase inhibition assay, and we found out that our strain SRK001, the crude extract of our strain, showed 27% inhibition. And we observed it by using positive control, clavulanic acid as positive control, which showed 48% inhibition. And we used nitrocephin as negative control, which showed 0% inhibition. Nitrocephin is a cephalosporin. After that, we from the crude extract, we separated the compound through TLC, thin layer chromatography. And after that, we did agar overlay TLC bioautography guided identification of beta-lactamase inhibitor using MRSA, that is a commercial strain. And the colorless zone in this bioautography, it indicates the inhibitory zone. Then from that, the inhibitory zone, the compound we purified the compound in reverse phase HPLC, and we observed multiple peaks. It showed that that is not a single compound, it is a mixture of our metabolites. So we have done till now, RPHPLC. In future, we will be doing single molecule isolation. We will be doing NMR spectroscopy, single molecule crystallography for novelty check. And we will also be doing LC-MS analysis for dereplication studies. Thank you.
Thank you, Divya. Now I ask audience to ask her question. Anyone?
It is? Yes, ma'am, it is a strain. We isolated.
You isolated it?
Yes, ma'am.
Okay,
From marine
it's a
sponge.
novel strain then.
Yes, sir. For novelty, we will be checking. We are in process.
Okay.
So this is specifically for gram-positive bacteria. Because you are using beta-lactamase. Beta-lactam is active on gram-positive bacteria, not gram-negative
Sir,
bacteria.
it is acting on gram-negative bacteria also.
Predominantly, it is for gram-positive bacteria, I guess, because that's why it is called narrow-spectrum antibiotics. So already there are a lot of antibiotics are in the market, which is a narrow-spectrum antibiotics. And actinomycetes is known for, antibiotic production, lot of antibiotic production. So why do you think that this beta-lactamase inhibitors is important for commercialization?
Sir, already in the market, there are many antibiotics available and resistance is shown against all the antibiotics. In third generation, fourth generation antibiotics, resistance is observed. So we are not discovering new antibiotics. We want to make an inhibitor which makes the existing antibiotics effective.
So you will apply it in combination with the existing.
Yes, it will show the synergistic effect with the antibiotic.
And did you try it with some kind of escape bacteria? Because escape are more prone towards.
Yes, sir. We are doing that.
Which is a burning issue that everybody is talking about, as you rightly mentioned in your first slide, that antimicrobial resistance.
Yes, sir.
Okay.
The strain I showed, it was MRSA Staphylococcus aureus. It was commercial strain, but we are currently in process. We have some escape strains, and we are performing our studies on that also.
Thank you.
Can you bring to that TLC slide?
Yes.
Can you explain these two figures?
Sir, the first figure, it is a TLC plate, silica gel TLC plate. Here we separated the compounds from the crude extract. We put the spot on this TLC plate and we perform the separation, and we observe multiple bands. The fluorescence, it is showing the multiple bands. Then we did TLC bioautography. We made the molten agar. We inoculated our commercial bacteria, which is antibiotic resistance. It is proven already. Then in that agar, we put beta-lactamase enzyme that was also commercially available. Plus, we put nitrocephin, that is a chromogenic substrate. So, in presence of nitrocephin and beta-lactamase, this plate showed red color. But, and yes, we poured this whole agar on the TLC plate. So, the compounds in the TLC plate, they diffuse in the agar. And then if the compound is showing inhibitory activity, the clear zone is observed.
My question is that RF 0.87, what is this value that your means compound of interest?
It's basically the distance traveled by
Not asking that thing. Okay. This is your compound of interest which you have marked RF
Yes, sir.
0.87.
Yes, sir.
And what is here? What is this RF where you have got this zone of inhibition kind of thing?
So this is the image of same plate, but after we perform the bioautography. This is, we poured the agar on this plate, TLC
You have
plate.
poured agar on this TLC
Yes,
plate
sir.
only.
Yes, sir. Then it showed the red color and it is shown clearless zone. That is why we confirmed that this band where we have written RF value 0.87, this compound is basically showing the inhibition.
Okay. Thank you. Thank you, Dibya.
Thank
Now I like to call next speaker, Survi from Sarda University.
This one for next and this is laser.
यहाँ से
ये बैक नहीं होगी
Good evening, everyone. I am Surbhi from a BTech Biotech third year student from Sarda University. Today, I am going to present my project on the topic of molecular identification of efflux pump resistant Klebsiella pneumonia, SBT003, isolated from human urine. The main aim of my project is the isolation and the characterization of the efflux pump mediated antibiotic resistance in Klebsiella pneumonia from human urine. The purpose of our project is firstly the screening of the efflux pump resistance microbial strain. Then we will done the biochemical characterization of efflux pump, and after that we have done the molecular identification of that strain. So first, let us know what is the antibiotic resistance mechanism. It is the biological phenomena of a bacteria which resists the antibiotic effects. It occurs when bacteria develops ways to survive the drug. There are primarily four major antibiotic resistance mechanisms. First is the efflux pump. Efflux pump is the actively transport channel, or I can say the pump which pump out the antibiotic before entering into the cell. Second one is the enzymatic degradation. In it, bacterial cell develop some chemical just like beta-lactamase, chloramphenol, acetyl transferase, something like that, which inhibit the entry of the antibiotic in the cell. Third one is the target modification, in which the cellular component mutated some like DNA gyrase, topoisomerase mutation, which also inhibit the antibiotic effect. And the third one is the reduced permeability, in which the bacterial changes the cell envelope to entry of the antibiotic. So since my objective focuses on the efflux pump resistance mediated by the efflux pump, so we will see now the efflux pump mechanism. Efflux pump are basically the energy driven by ATP or PMF. So these, as you can see in my slide, these are the steps which the efflux pump works. Firstly, the antibiotic enters to the cell, then the bacterial efflux identify it and expel out through the membrane. The decreased concentration in the bacterial cell decide that the antibiotic in the cell is less due to the efflux pump activity, or maybe it can be some other activity just like we seen in the enzyme degradation or the target modification. Since efflux pump is a broad spectrum antibiotic resistance mechanism, it can not only resist the one antibiotic, it can resist a different class of antibiotics such like the fluoroquinolones and tetracycline, cefin, all of this. So it is also considered as the multi-drug resistance mechanism. Now we move towards the methodology. Firstly, we collected the sample. The sample was collected from a UTI infected patients of human serum. Then we stick the plate of the bacteria, isolated the plate bacteria on an agar plate, and then we perform a series of the biological testing. Firstly, gram-negative urease, citrate, indole, and methylate. As you can see in my table, that gram-negative is, that gram is staining, it is a gram-negative bacteria. It shows the positive result for urine, positive for citrate, and indole and methyl, it shows the negative result. After that, we performed the antibiotic susceptibility test. It determined the sensitivity and resistance of the isolated strain SBT003 against the commonly used antibiotic present in the market. For this, we use the disc diffusion method. We place the antibiotic disc on the inoculated agar and measure the zone of inhibition. And for this, we also use the Vitek instrument for the antimicrobial profiling. But from the result of Vitek, we get to know that the bacteria is resistant to the levofloxacin, ampicillin, cefin, and tetracycline, and all the other antibiotics. It is sensitive. This show that the efflux pump is all the antibiotic which the bacteria or resistance is a hallmark of the efflux pump activity. That's why it's proved that our bacteria is a Klebsiella pneumonia. Till now, we have done all the phenotypic characterization of our bacteria. Now we move towards the molecular identification, which we do by the 16S rRNA sequencing and using PCR. For this, we isolated the genome, done the amplification process with the 16S rRNA primer, and in these slides, you will see that the PCR's amplicons that we have highlighted in the red box. Then we perform the blast analysis result. From blast analysis, we get to know that the stain have the 100% similarity with the previously stain derived, which the Klebsiella pneumonia stain NKH21, and I have also attached the sequence alignment photos. For evolutionary relationship, we done the phylogenetic analysis and the chromatogram, which suggests the sequence genomic sequence. In phylogenetic analysis, we also observed that our bacterial strain have a various evolutionary relationship of the previously isolated strains. So we concluded that, we successfully isolated and molecularly identified our strain, which is the Klebsiella pneumonia strain SBT003. And our future work focuses on the whole genome and exome sequence analysis, comprehensive characterization of antibiotic resistance genes, which is basically the ACR, AB, TOL-C, or the OQX-AB. And then we are planning to perform the bioinformative and the comparative genomic studies of that gene. Thank you, sir.
Thank you, Survi. Now Survi is open for question from the participants or from the audience. Anyone? Survi, I just want to know, since you are BTech student, I'm not going to ask a lot of questions. Just I want to ask, have you done this by yourself only?
No, sir, I have a proper team. Means I have a 4-member team. I'm representing all of them.
What work you have done?
Sorry?
What work you have done out of this work zone?
We perform wholly, not by single.
Okay. Very good one, Survi. I like your courage to come in the BTech third year and present at this level. Good, keep it up. Thank you.
Thank you, sir.
Next speaker is Renu Bala from Amity University. Renu Bala from Amity University. The topic is hypertension among the Gaddi tribe. A study from Chamba district of Himachal Pradesh.
This one for next time, this is Nicholas.
Okay, I don't. Yes. Good evening, everybody. I am Renu Verma, and I am pursuing my PhD from MIT University, Noida, from the Department of Anthropology under the supervision of Dr. Professor Rumi Dev. I have completed my pilot study, and based on that pilot study, today I am presenting this work. And the title of my work is Hypertension Among the Gaddi Tribe, a qualitative pilot study from Chamba district of Himachal Pradesh. Talking about my introduction, I just mentioned some paper who reveals the prevalence of hypertension globally and in India, and as well as in Himachal Pradesh. So hypertension is widely prevalent disorder which affecting 1 million people globally, and WHO 2020 reports states that approximately 1.28 billion adults of age group 30 to 79 years living with the hypertension. Among them, 46 are unaware, while 42% are getting treatment, and only 21% achieve the control. According to the study of global burden disease 2019, estimates that 1/5 of global deaths occur due to the elevated pressure, and it is the major health issue in India as well as in global. So study from India over the past 2 decades show the rising hypertension in both urban and rural population with high prevalence report across the region. So study of Gupta et al. Mentioned that the poor control in developing countries like India is due to multiple factors such as behavioral, social, and healthcare access factors. So hypertension is rising among India tribal population due to lifestyle transition, dietary change, and acculturation. While the tribal population once believed that they are protected from this disease because they led hyperactive lifestyle and traditional dietary habits. According to a study of Rena, the Gaddi tribe residing in Kangra and Chamba district now experiencing high risk of hypertension and its risk factors like cardiovascular disease, just because of rapid modernization and change in dietary habits and lifestyle factors. So hypertension prevalence in Gaddi tribe is 10.5%, which is lower than 36% in non-tribal Himachali population, but indicates an emerging risk of hypertension and its risk factors like cardiovascular disease. So rationale of my study is rational and objective of my study is the Gaddi tribe is geographically isolated and socio-culturally distinct population with limited epidemiological data on hypertension and cardiometabolic risk. In view of this evidence gap, the study aims to assess hypertension status and associated cardiometabolic risk, including general and central obesity. The study further evaluates health-seeking behavior, quality of life, and related knowledge, attitude, practice, and behavior. The assessment is conceptualized within ongoing lifestyle and dietary transition that may be contributing to an increasing disease burden in the community. Method and methodology. So this study is cross-sectional study conducted as a part of an individual PhD research project. Studies conducted in Gaddi tribal villages of Chamba district, Himachal Pradesh, with the population of 20 to 60 years, and the sample size was 60 because it was pilot study, and the research tool was standard schedule interview designed with an option for qualitative open-ended and qualitative closed-ended for data collection. Result of my study, the total of 60 Gaddi adults participated. Among them, 27 were men and 33 were women of age group 20 to 60, and mean age was 46. Hypertension was identified in 12 participants. Among them, 5 were men and 7 were women. So hypertension participants commonly showed higher BMI, that is body mass index, and waist hip ratio among women, and hypertension was mainly seen in older age group and was linked to reduced physical activity, post-menopausal status, higher BMI, and limited healthcare access. Talking about the knowledge, lifestyle, health-seeking behavior of Gaddi community, the Gaddi community have very limited knowledge about the hypertension. For them, hypertension is very normal, and they perceive it no serious illness. In my structured interview and focus group discussion, when I ask them some question, they respond like, ये तो कोई बहुत बड़ी बीमारी नहीं है, इसके लिए कोई घबराने की ज़रूरत नहीं होती, और एज के साथ तो ये बीमारी आती ही आती है, तो इसके लिए हम कोई कुछ भी सावधानियां नहीं ले सकते, और लेंगे भी तो आएगी ये बीमारी. टॉक अबाउट द एटीट्यूड, दे सेड कि अगर हम दवाई लेते हैं तो 2-3 दिन में हम ठीक हो जाते हैं और उसके बाद हम मेडिसिन नहीं लेते हैं. सो हम सर बीपी की बीमारी से हमें कुछ भी ऐसी कोई जानलेवा डर नहीं है कि हमें कोई जानलेवा प्रॉब्लम हो जाएगी. And most of male participants, when detected with hypertension, they reacted and they just leave the place and they said कि हमें बीपी की कोई बीमारी नहीं है, हमने रात को शराब पी थी इसलिए ये हमें बीमारी, इसलिए हमारा बीपी बढ़ गया होगा. एंड मोस्ट पार्टिसिपेंट सेड दैट कि हम तो बहुत अच्छे से काम कर लेते हैं, हमें कोई प्रॉब्लम नहीं है, हमें कोई सिम्टम नहीं आए, बिकॉज गद्दी कम्युनिटी रिलेट द एसोसिएट हाइपरटेंशन विद सिम्टम लाइक dizziness and headache and vomiting, and most the participants said कि हमें तो ऐसा कुछ नहीं हुआ, which detected with hypertension during our survey. So they said कि हमें तो ऐसी कोई प्रॉब्लम नहीं है, आप हमें वहम डाल रहे हो आके. And mostly I find Gaddi community male dominating society because they were not comfortable with me to give their BP measurement to me, while they are comfortable with the male coordinator who was with me in my survey. And yeah, female participant was very comfortable, and they were participated actively, and they give their measurement. And if they detected with hypertension, then they asked me that what precaution they can have. Older adults reported reduced physical activity. Younger adults, hard physical active lifestyle, alcohol use, and frequent meat intake common among hypertension adults. Most participants reported seeking care primarily from government health centers and district hospitals. When I asked them why don't they go to the private hospital, the female participant said that there is no need to go to the private hospital. Like they respond like कि हमारे पास इतना पैसा नहीं है कि हम BP की बीमारी
Ma'am, wrap it in 1 minute, please. Due to time
Key
constraint.
factor contributing to hypertension. Hypertension was mainly observed among adults aged 50 to 60 years. Older adults reported reduced physical activity, while younger adults followed a more active lifestyle. Regular alcohol consumption was commonly reported among hypertension participants. Frequent meat consumption was more common among adults with hypertension. Only 3 out of 12 hypertensive participants reported regular adherence to medicine, distance to healthcare facility, medical shops, contributing to regular treatment and follow-up. So concluding my study, the present study demonstrates a substantial burden of hypertension among Gaddi tribal adults residing in high altitude region of Chamba district of Himachal Pradesh. Despite geographic isolation, traditionally active lifestyle, hypertension was common and frequently under-recognized. Awareness was limited, health-seeking behavior was largely symptom-driven, and regular monitoring practice were uncommon. These findings indicate an ongoing epidemiological transition within this tribal community and underscore the need for culturally appropriate screening, early detection, and sustained community-based intervention to improve awareness and blood pressure control in high-altitude tribal population. Thank you all. This is all for my work and presentation.
Thank you, Renu Bala. Any question from the participants?
Hi. Actually, I am from Chamba district. So I'm just wondering why you chose Chamba for this, because we always consider Himachal is a peace-loving and, a healthy persons there. And yeah, of course. So I'm just wondering why you chose Chamba for this.
I'm also myself from Kangra district.
So
It's a
why you
neighboring-
chose for
Yeah,
this? You
because-
should
yeah,
have
yeah.
you should have a data from the NCR, the metropolitan city, because here we are suffering from such kind of disease, hypertension and heart disease and
But
all
I
these
think
things.
here people are more aware about the hypertension. They have access to healthcare facilities. And being a Pahadi, I know that the
I don't think so,
tribal
because
population have low access, very zero access of healthcare facilities, and very limited. I think zero awareness about the hypertension, because I have studied them for 1 month, and they taken like it's not for Chamba people, it's for Bharmor people. I just went to Bharmor, and if you are from you are from Chamba, and you know that Bharmor is very interior, and like it's very difficult to access that area, and moreover very difficult to access the people, because they are not ready to give. If I'm say, the pradhan of the village introduced me like a medical staff, a doctor came from medical staff. He didn't introduce me like a researcher, because people don't give any kind of qualitative and quantitative data to the researchers. And I find key people are zero knowledge about hypertension, while they are living in very protective
environment,
atmosphere. In spite that they are suffering with high prevalence of hypertension. And moreover, I just wanted to convey one more message. The tribal women, more than hypertension, are suffering with the other disease. They more female participants reported that they have had severe body ache, severe joint pains, particularly in winters. They have severe swelling in their joints of hands and feet. Okay, so thank you.
Thank you, Renu. Just suggestion. It will be better if you do such kind of study in NCR or in Uttar Pradesh, people who are doing academic job and how they are influencing this academic load is influencing hypertension in these people. Okay, I'm very much interested. I think it's a very good study, and thank you, Renu.
Thank you all. Thank you for listening me.
Thank you all. With this, we conclude today's session. But before concluding, we would like to felicitate our esteemed session chairs. And for this, I would like to invite Dr. Amit Chaurasia, our session in charge, Dr. Guru Murthy, me, myself, and also Dr. Preeti Mehta Kakkar, my partner, to felicitate our kind esteemed chairs. So first, we like to invite Dr. Ranjan. Dr.
And Dr. Shera. Dr. Rajesh Kanal. May I request Dr. Rajesh Kanal? Thank you so much, sir. Now may I request Dr. Shera to please join us for the facilitation. So on behalf of Amity University, Uttar Pradesh, and the organizing committee of International Conference of Sustainable Biotechnology and Bioinnovation, we would like to express our sincere gratitude to our respected session chairs for their leadership and academic insight, to all our speakers for sharing their cutting-edge research, to our faculty members, researchers, and students for their enthusiastic participation and engaging discussion. Our heartfelt thanks to the organizing committee and our collaborative partners, the Biotech Research Society of India, International Bioprocessing Association, and the Center for Energy Environmental Sustainability for making this session a great success. We would also like to extend our sincere thanks to our gifting partners, Unipic and our youth for their continued support. With this, we formally conclude the proceeding of day 2 in E2 Seminar Hall. Thank you so much to all. कौन सा पे है? Faculty F1 launch, F2 and F1. So all are requested to join us for a tea session for the faculties F1 launch, and for the students F2 backside. Thank you so much.
Can I close now?